All right. Hi, everyone. Wait. Yes, please. All right. Good evening, everyone. Welcome, guys. Welcome back to the NEET English channel, Unacademy NEET English channel. Hanji. Good evening. Good evening, all of you. Just a second. Quickly join the class and please bring your NCRTs with me. Share the channel, share the session with everyone. And guys, give me a second. Quickly. Shh. Give me 1 second. Alright guys, welcome back to the channel. Good evening all of you. How are you all? Yes, please. Now we are all set. I hope everybody is joined in. Good evening all of you. The class has already started. Good evening all of you guys. So as you know, we have completed class 11th biology yesterday, except for 1 or 2 chapters, which I'll be discussing today along with this class 12, because we have lesser syllabus today to complete. So as yesterday we started from the backwards. So we are going to start from chapter 13, that is ecology. And also the new trend has been that NCERT NTA has started giving more questions from ecology. Last year we got around 10 questions from ecology, right? 10 questions. So let's give it more importance and let's start with this unit. All right. NCERT is ready. Give me a thumbs up that we are all set with NCERT and we can start with the session. Again, running back to front, yes. Okay, I'll tell you one very silly superstition that I have. I feel that whenever I have to quickly finish the syllabus, it's something that I have been practicing since my school days. So whenever I start finishing my revision from backwards, I'm able to finish that off quickly. I don't know. There is no logic behind it. No logic at all. It's the most silly and the illogical thing that I practice, but I feel my work gets done easily and quickly. Anyways, good evening, all of you. Let's get started. Perfect. Now, starting with chapter 13, biodiversity and conservation. As I've told you, NTA is giving more questions from ecology these days. So we are going to start with this chapter. The first line that is important and you must highlight the most, that there are more than 20,000 species of ants, 3 lakh species of beetle, 28,000 species of fishes, and nearly 20,000 species of orchids. Not very important line, I'm sorry. But yes, you should do that once. Okay, moving on to the next line, that is, the important thing is biodiversity. This term biodiversity was popularized by a sociobiologist named Edward Wilson. You must know his name. Timings of these sessions, we have just started. Otherwise, guys, your unit-wise revision will continue, as I've already told you, from Monday. This time we won't have class on Wednesday. We'll have it Monday and Tuesday. So we'll have regular class so that we can complete with our unit wise revision. So detailed lectures will be uploaded. Okay, and the timings are 5 p.m. So those of you who have joined me in this batch, in this session only, they must know my usual classes are at 5 p.m. Now coming on to the next, genetic diversity. Okay, now we'll concentrate here. I won't be reading your chat for a while. Okay, after completing this chapter, I'll see your chat. Now see, genetic diversity, the important lines that you must know of, that single species. What is genetic diversity? In this, single species might show high diversity, and the medical plant, for example, Rauwolfia vomitoria, we have so many species, so many genetic diversities of this plant based on the potency and the concentration of this chemical reserpine. So you should know that Rauwolfia vomitoria has this chemical known as reserpine. And similarly, we have 50,000 genetically different strains of rice and 1,000 varieties of mango. Now, these values are important. Is that clear? 50,000 strains of rice and 1,000 varieties of mango in India itself. Okay, let's not talk about the world here. In India itself, there are so many. Then species diversity, if we say, then Western Ghat has greater species of amphibians. Then ecological diversity means in India we have so many ecosystems, for example deserts, rains, and so on, and India has greater biodiversity than Scandinavian country like Norway. Then coming on to this very important abbreviation IUCN, the data that we usually quote, the full form for IUCN is International Union for Conservation of Nature and Natural Resources, and the total number, according to IUCN, the total number of plants and animals which have been described so far is slightly more than 1.5 million. You should know this. Now, according to the extreme estimates, there are around 20 to 50 million species. But according to Robert May, the global species diversity is around 7 million. Are these points clear? Anyone with any doubt? Then moving on to the next page. So, out of the total Earth's biodiversity, 70% diversity is of animals, and plants are not more than 22%. And when we say plants, it includes all algae, fungi, bryophytes, gymnosperms, and angiosperms. Are these points clear to all? Right? Then out of 70%, which are animals, now out of those 70% animals, more than 70% are insects. That means out of every 10 animals on this planet 7 are insects. Are these points clear? Now these graphs are very, very important. You should be able to identify the labelings here, so don't ignore these graphs. Is it clear? So see the labelings. Now coming on to the next page, see here we have a lot of data. India has only 2.4% of the world's land area, but according to the global species diversity, we have around 8.1%. Now India is also one of the 12 mega diversities. There are 12 mega diversity countries in the world, but India is one of those. Now nearly 45,000 of species of plants are known, and twice, that means 90,000 species of animals have been recorded from India. Now according to Robert May's global estimates, 22% of the total species have been recorded so far. So that means rest of 78% are not even recorded till now. Are these points clear? Okay, moving ahead, yet they are yet to be discovered. So more than 1 lakh plant species and more than 3 lakh animal species yet to be discovered. See, I understand that these data may not be, they may not sound very interesting and very important, but you never know. Just learn important data. I'm not going to ask you to learn all the data here. That may be time wastage, but let's learn some important data. Let's choose. Now coming on to the patterns of biodiversity. So we see that there is species diversity more towards the equator. So this line says that species diversity decreases as we move from equator towards the poles. So that is why there are more species of birds observed in Colombia, because Colombia is near the equator. Similarly, New York has 105 species. Don't learn it, just learn the pattern. Colombia is nearer to equator, so more diversities. Then New York, so lesser than that. Greenland is even towards the pole region, so even lesser. So India has around 1200, because India is not at equator but near to the equator, near to the Tropic of Cancer. Is that clear? Near to the Tropic of Cancer, right? Then Ecuador. Ecuador has 10 times more species of vascular plants than the equal area which has in temperate region like in Midwest. Amazon rainforest in South America has the greatest biodiversity because we know it is an equatorial region. And the more you are near to the equator, the more is the the species diversity. Are these points clear? Are these points clear? I'll give you mnemonics wherever they are important. Is it clear? Okay. Now just learn the trend. You don't even have to learn the number. You just have to learn the trend and then you can just match it as per the decreasing order. Is that clear? Then what is the reason behind tropics having the greater biological diversity? See, this is important. So the reason number one is that species is generally a function of time. You can get this in the statement format. So species is generally a function of time. Now, because temperate region has been subjected to a lot of glaciation, therefore tropical regions have remained undisturbed. So undisturbed being undisturbed means more biodiversity. Then tropical environments are less seasonal and relatively more constant. So being more constant, it promotes the niche specialization. Third reason is that tropics have more solar energy, right? So since there is more solar energy, there will be higher productivity and greater diversity. Then very, very important. This graph is very, very important. It has already come in PYQ about species area relationship. Now this relationship was explained by a geographer, German naturalist and geographer Alexander von Humboldt, where he says that within a region, species richness increases with increasing explored area. That means if you increase the area, the number of species will also increase, but only up to a limit. We will discuss everything. We'll complete class 12 today. Don't worry. Yes. So now this equation, see the straight line that we see here, this is in terms of logarithm. But the blue line in the graph, see it is a rectangular hyperbola. So first it increases and then it doesn't increase. So there is a limit to it. So the equation is S is equal to C A raised to the power Z. So what is S here? You should know all the elements here. So S is the species area, C is the y-intercept, and A is the area, and Z is the slope. Is that clear? Z is the slope. So if I take the log, it is log S is equal to log C plus Z log A. Now you should know all these elements. Z is the slope, or we call it the regression coefficient. What is the usual value of Z? Regardless of taxonomic group. See, now you have to be careful here. If we are not considering any taxonomic group here, then the general value for slope is 0.1 to 0.2. Is this clear? But for the entire continent, this value decreases. Sorry, increases to 0.6 to 1.2. And for frugivorous, that is fruit-eating birds and mammals in the tropical forest, this value increases further to 1.15. Are these points clear to all? I hope you are underlining. I hope you are underlining all the important lines here. Is this clear? All right. Now, what is a stable community? Very, very important. What are the features of a stable community? The community which does not show too much of variation in productivity from year to year. That is one. Second, it should be resistant or resilient. Resistant means either it should not occur. Resilient means even if the disturbance occurs, then it is able to recover back. So, a community should be resistant or resilient to the occasional disturbances, man-made or natural. Third point for a third feature of the stable community is that it should be resistant to the invasions by alien species. Are these points clear to all? These are all features of stable community. Then now these scientists and their contributions are the most important thing in this chapter. So let's talk about David Tillman. What does David Tillman say? David Tillman said that long term he performed long term ecosystem experiments in the outdoor plots. And what did he observe? He found that the plots with more species showed less year to year variation in the total biomass. So basically this is in reference to the stable community. So what did he say? That plots which have more species are a stable community, right? And increased diversity means higher productivity. Are these points clear to everyone? Please let me know. Now coming on to the second experiment here. This is by Stanford ecologist Paul Ehrlich. So he says he gave us this rivet popper hypothesis. Now very, very important. What does Paul Ehrlich say? That in an airplane, all the parts are joined by small rivets. So basically now the question comes like this, that in Paul Ehrlich a rivet popper hypothesis of Paul Ehrlich, what is airplane equivalent to? So airplane, you must know, is equivalent to the ecosystem, while see here, while here the rivet is equivalent to the species. Are these points clear to everyone? Rivet is equivalent to species, airplane is equivalent to ecosystem, and the flight safety is equivalent to proper functioning of the ecosystem. So if you remove one rivet, it may not affect the flight safety, but similarly if we go on removing one by one the rivets one by one, then the flight safety will of course be compromised. So loss of rivets on the wings, loss of rivets on the wings is obviously a more serious threat. That means it depends which species or which rivet is being removed. Are these points clear? Are these points clear to all? Please let me know. Please let me know that these points are clear to everyone. Shall we move on? Okay, so we are already done with stable community. I've told you this rivet popper hypothesis. Now coming on to the loss of wait. Yes, talking about loss of biodiversity. So, a loss of biodiversity, see here, loss of biodiversity, guys. Just a second. I'll be here. Okay, so we are talking about loss of biodiversity. So, loss of biodiversity, how much loss of biodiversity has already occurred? This has been quoted in IUCN Red List. So, Red List, the data we use is from 2004. So, according to 2004 Red List, the recent see, there are 784 species which have gone extinct. Now, the main question comes from the recent extinctions in the match the following. You've already gotten this question in your PYQ. Very important. Dodo got extinct from Mauritius. Kaga got extinct from Africa. You have to learn the name of the organism and the country that it got extinct from. Please see here. Please see here. Now, thylacine is was from Australia. Stellar sea cow was from Russia, and three subspecies of tiger. The name of the subspecies was Bali, Javan, and Caspian. These were the recent extinctions. So, overall, in the last 20 years, 27 species have undergone extinction. Clear? Presently, 12% of all the bird species, 23% of all mammals, 32% of amphibians, and 31% of gymnosperms are facing the threat of extinction. Now, presently, facing the we are witnessing the sixth extinction. And how is this sixth extinction different from the previous five episodes of extinction? That is, it is 100 to 1000 times faster. How fast is it? It is 100 to 1000 times faster. Are these points clear? Everyone, please let me know. Are these points clear to everyone? Now, what are the causes of biodiversity loss? Very important topic. So, the four major causes are collectively known as evil quartet. This is a PYQ. Now, out of these four major causes, habitat loss and fragmentation is the most important cause. Another PYQ. Now, this habitat loss and fragmentation can be easily seen or watched in tropical rainforest. So, earlier it used to have 14% of the total earth area, which now is not even more than 6%. Now, Amazon forests are called the lungs of the planet, but now why are they facing this reduction in the area? Because the land is being cleared for cultivating soya bean and for converting the grassland for raising the beef cattle. So, these are the two reasons that you must know. These are the two main reasons that you must know of, that why Amazon is facing loss of area and therefore habitat loss. Second cause of biodiversity extinction is over exploitation. So again, last 500 years, some species which have undergone extinction are stellar sea cow and passenger's pigeon. Then alien species invasion. When the alien species come to the native area and disturb the native diversity. So example is Nile perch, introduced in the Lake Victoria. It caused extinction of 200 species of cichlid fish in the lake. Is this clear? Then similarly, we have another example of alien species. So alien weed species, carrot grass or parthenium. lantana, water hyacinth or ishornia. All these three are the examples of alien species, but now they are so common around us. Then third example of alien species invasion is introduction of African catfish known as Clarius gariepinus, and it was a threat to the indigenous catfishes in our rivers. Are these points clear to all? Please let me know. Please let me know. So guys, next is co-extinction. Let's talk about the co-extinction here. So co-extinction means when the two species which are bound to each other in the obligatory manner, right? For example, in symbiotic association, so or they are they are obligatory to each other, they always show co-extinction. They evolve, they show co-evolution, and they show co-extinction. For example, if the host dies, the parasite also goes extinct. Similarly, if a plant on which the pollinator depends, plants die, the pollinator also goes extinct, and the vice versa. Are these points clear to everyone? Please let me know. Now, what are the reasons that why we should conserve biodiversity? So there are three categories, three viewpoints. Now, what is the narrowly utilitarian viewpoint? Very, very important. Narrowly utilitarian argument says because we are getting countless direct economic benefits from the ecosystem, that is why we have to conserve it. Now, what are the direct economic benefits? We get foods and firewood, fiber, construction material, other industrial products, and medicinal products of medicinal importance. Now, this line is important. More than 25% of the drugs which are derived from the plants, drugs that are sold in the market, more than 25% of the drugs sold in the market are derived from plants, and 25,000 species of plants contribute towards the traditional medicines. Are these points clear? So we have to preserve them, we have to conserve them. Now, see this word bioprospecting. Now we should be able to understand how NTA works, how NTA just surprises us. Now see here, there is this word bioprospecting. I get a very strong feeling that NTA might ask this question, that what is bioprospecting? It is exploring the molecular, genetic, and the species level diversity for products of economic importance. I think NTA can ask this question in 2026. So what is bioprospecting? Is that clear? Then about the broadly utilitarian argument, the broadly utilitarian argument says that biodiversity should be conserved because ecosystem services are provided. Now, what are the ecosystem services being provided here? First of all, it gives us oxygen. Second service is pollination. And third service is giving us aesthetic pleasures of walking through the thick woods, watching spring flowers. All these are examples of this. These are the four examples of the ecosystem services. Now, third is the ethical argument. Ethical argument is very philosophical in nature, which says this globe belongs to all the different species. We are not the only one species, and each species has an intrinsic value. It is a moral duty. So all that morality and that philosophy comes in this ethical argument. Are these points clear? Now, very, very important from this. This is the third most important topic from this chapter. See, first is the causes of destruction, right? Causes of loss of diversity. Then second is this conservation, and third was the Alexander's that species richness graph also. Now talking about the methods of conservation. First is, guys, in situ conservation. Now what is in situ conservation? When we protect the organism in its own homeland, in its own habitat. So we don't have to take it away from the place of danger. Now see, in situ conservation, what are the things that come in? Number one, the methods of in situ conservations are biosphere reserves, making national parks and sanctuaries. And then fourth is sacred groves. These are the four methods of in situ conservation. How many biosphere reserves do we have in India? 14, 90 national parks and 448 wildlife sanctuaries. Then sacred groves, where are these found? So you must know the location. You can get it for match the following. Is that clear? Also, now, this is very, very important. Biodiversity hotspots. Which area is designated as hotspot? The hotspot has to have these two characteristics. Number one, the region should have species richness. So there should be a huge biodiversity. different type of diversity. So species richness should be there. And second, it should have certain species which are only confined to that region. That means high degree of endemism should be found. Is that clear? So any area, any region which has these two characteristics may be regarded as a hotspot. Earlier we used to have 25 hotspots all over the globe. Now 9 were added. So in total, in today's date, we have 34 hotspots. So another characteristic of hotspots these days is that these regions are facing accelerated habitat loss. Are these points clear? That India comes under three hotspots: Western Ghats, Sri Lanka, Western Ghats and Sri Lanka, Indo-Burma, and then Himalaya. Is it clear? Now, surprisingly, all the hotspots put together, they cover only 2% of the land's area. Are these points clear? Again, I'm revising the methods of in situ conservation: biosphere reserves, national parks, sanctuaries, and sacred groves. Then we have ex situ, where the organism is taken out of their natural habitat. Now, the methods are zoological parks, which are easily known as zoos, botanical gardens, and wildlife safari parks. Also, fourth example is that of seed banks, pollen banks. These are all ex situ conservation methods. Now, the two summits that have already taken place: the Earth Summit in 1992 held in Rio de Janeiro. Now, the objective was that all nations have to take appropriate measures for the conservation of biodiversity and sustainable utilization of its benefits. So, Earth Summit happened earlier. Then, in 2002, happened the World Summit on Sustainable Development. Where did it happen? It happened in Johannesburg in South Africa. So 190 countries pledged their commitment to achieve by 2010, which is already gone by actually, a significant reduction in the current rate of biodiversity loss at global, regional, and local levels. Now, please let me know that all these points are clear to you. Yes, guys, please let me know all these points are clear to you. Have we completed this first chapter? How many of you have completed this chapter with me? How many of you guys have completed this chapter with me? Done? Only 4 of you? Me? No, Bablu, we can't be slow. You already know we can't be slow here. Alright guys, moving on to the next chapter, that is ecosystem. First line, ecosystem is a functional unit of nature. Underline. Now, there are different types of ecosystems. We have terrestrial ecosystem. Sorry. So terrestrial ecosystem is represented, the examples are forest, grassland, and desert, while pond, lake, wetland, river, and estuaries are the example of aquatic ecosystems. Then similarly, there are man-made and then there are natural. So example of man-made ecosystem is crop field and aquarium. I hope this is clear. Now, coming on to the next page, this definition: vertical distribution of different species occupying different levels is called stratification. So vertical distribution. So if we go from ground to the above, so what do we see? That trees occupy the topmost strata, right? And similarly, herbs and grasses will hold the bottom level. So the components of ecosystem, please see this. What are the 4 components of ecosystem? Ecosystem should show productivity. Then we talk about decomposition, energy flow, and nutrient cycling. See, out of these 4 components, nutrient cycling is not in your syllabus anymore, but we have to talk about the rest of the 3. Now, see, ecosystem, any ecosystem is a fairly self-sustainable unit. Please underline these lines and remember them. Now, autotrophic organisms in an ecosystem are, what is the example of autotrophic? Phytoplankton, some algae, floating, submerged, and marginal plants found on the edges. So all those which are photosynthetic are autotrophic. So these are the examples of autotrophic. Similarly, consumers, which depend on these autotrophs directly or indirectly. So consumers, the examples are zooplankton, free swimming and bottom dwelling forms. Then third are the decomposers, which is fungi, bacteria, and some flagellates. So we have seen the examples of autotrophs, consumers, and the decomposers. These are the main components of any living part of the ecosystem. Now, another line: unidirectional movement of energy will flow from from lower trophic level to the higher trophic level, and its dissipation as a loss of heat to the environment. This will also occur. Very, 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 very important. The first most important thing from this chapter is the definition of and difference of primary production and productivity. See, you usually get to identify what is the correct unit of production and productivity. Now please watch it carefully. What is the definition of primary production? Amount of biomass and organic matter produced per unit area over the time period by plants during photosynthesis. Now it is expressed in terms of weight, that is gram per meter square, or energy like kilocalories per meter square. Now this is production, but now when we talk about production with respect to time, it becomes productivity. See, what is the definition of productivity? Rate of biomass production. And we bring time factor in this. Whenever there is word rate, we are bringing time factor to it. So see the change in the units here. So production was gram per meter square. Here productivity is gram per meter square per year. Similarly, in terms of energy, production was kilocalories per meter square, but productivity is kilocalories per meter square per year. One very, very important. Aren't you coming now? Never mind. You just go with the flow. We are reading NCERT class 12 today. I've completed only one chapter. You can read that chapter later on. Okay, no problem at all. All right, guys. Moving ahead. Now this is the first very, very important topic that I've discussed with you from this chapter. Please note. And similarly, what is gross primary productivity? It is the rate of production of organic matter during photosynthesis, of course, by the plants. Now, what is net primary productivity? It is the available biomass of consumption, available biomass for the consumption of the heterotrophs. So what plants are producing is gross primary productivity, and what is left over for the next trophic levels, that is the for the heterotrophs, is known as the net primary productivity. Now what is the formula? So net primary productivity is always less than gross primary productivity. Why? Because of some respiration losses. So GPP minus respiratory losses is equal to NPP. Are these points clear? Slow karo na ma'am. How can I go slow? That we have to complete the entire NCERT book today, and also few chapters of class 11. Okay, let me know one thing. How many of you have taken the mock test from 2 to 5? How many of you have taken the mock test from 2 to 5? Hi Charu, Peksha, good evening. Yes, how many of you have taken the mock test today? Tarun took it. That's it. So guys, those of you who have missed today's mock test, you have to enroll for it. The link is given in the chat. You have to take it every day. You have to take it every day. Now till your final exam, you have to practice that thing every day. Go in that zone of paper. You were attending physics class live that time. You can always watch it recorded, isn't it? But don't, spare that time, 2 to 5, for your exam, for your mock test now. Is that clear? All right, moving on. Moving on to the net primary productivity, I've already told you. Then we have secondary productivity. It is the rate of formation of new organic matter by the consumers. Now see the values. The annual net primary productivity of the whole biosphere, that is match the following. Annual net primary productivity of the whole biosphere is 170 billion tons. That is the dry weight. And then 70% of the surface. So out of this 170 billion tons, despite occupying 70% of the surface, the productivity of oceans is only 55 billion tons. So can you tell me what is the reason behind that? Can you tell me why the productivity of oceans is 55 billion tons only out of the total 170 billion? Anyone who can answer me? Anyone who can answer me? And then coming on to second very, very important aspect, that is the process of decomposition. So answer to this question is, guys, because there is less penetration of light inside the ocean. So less solar input means less productivity. Is it clear? Less population, no, less input of solar energy. Now, moving on, yes, less sunlight reaches to them. Now, moving on to this topic, decomposition, very, very important. What is the definition of decomposition? breaking down of complex organic matter. See, breaking down of complex organic matter into the organic substances, simple organic substances, is called decomposition. Now, decomposition occurs in some steps. So there are 5 steps involved in decomposition. It is fragmentation, second is leaching, third is catabolism, then we have humidification, and fifth is mineralization. So you have to learn all these 5 steps. So what happens really? So first of all, anything which will undergo decomposition, that means a raw material for decomposition, we call it detritus. So raw material for decomposition is known as detritus. So it includes dead plants like leaves, barks, flowers, dead remains of animals, fecal matter. All this is detritus. So how does it undergo decomposition? So first of all, see, 3 things are going to happen simultaneously. So first 3 steps, fragmentation, leaching and catabolism, they are going to take place simultaneously. Are these points clear? So see what happens in fragmentation. So there are certain organisms known as detrivores. For example, earthworm, it breaks down the detritus into small particles. So from large particle to small particle, this is fragmentation done by detrivores. Is it clear? This is the first thing. Simultaneously, leaching is taking place. So what is leaching? So the compounds or the water soluble inorganic nutrients which are present, they simply get leached, they simply get precipitated into the lower zones of soil, and they precipitate there as unavailable salts. So this is known as leaching. So while fragmentation is occurring and leaching is taking place, simultaneously there is bacteria and fungi degrading the detritus using their enzymes into simple inorganic substances. So this is known as catabolism. Are you getting it? Earthworm is playing its role by breaking it into smaller pieces, fragmentation. Second, the water soluble inorganic nutrients present in the detritus simply get precipitated, no further action. Third, the bacteria and fungi is acting over the detritus also. So if bacteria and fungi is degrading the detritus using their enzymes being saprotrophs, this is known as catabolism. Is this clear? Then after catabolism, humidification happens. Humidification takes place. So what is humidification, guys? In humidification, there is accumulation of dark colored amorphous substance known as humus. Is this clear? So humus formation is called humidification. Now remember the characteristics of humus. Number one, it is dark. It is clearly written. It is dark colored. Okay. Second is amorphous. Amorphous means it doesn't have a specific structure. It is not crystalline. Third, highly resistant to microbial action. Fourth, decomposition is very slow in humus. Fifth, it is colloidal in nature. And sixth, it is a reserve of nutrients. Are these points clear? So there are 6 features of humus, 6 characteristics of humus that you must remember. Is that clear to all? Is that clear to all? Then, moving on to the next. Okay. See here. Moving on to the next part. So after humidification takes place mineralization. So what is mineralization? The humus which is formed is further degraded by some different microbes, and they will release inorganic nutrients during that process. So release of inorganic nutrients, that means minerals, is known as mineralization. Now there are certain factors which affect this process. It is an oxygen requiring process, first of all. Second, it depends on the rate of decomposition, will depend upon the chemical composition also. So for example, if detritus has large amount of lignin and chitin, then slow decomposition. But if detritus is rich in nitrogen and water soluble substances like sugars, then quick decomposition will take place. Third factor is temperature and soil moisture. So more the temperature, more the moisture, more is the decomposition. See, warm and moist environment favors decomposition, while low temperature and anaerobiosis, that means low oxygen, these two conditions, they will inhibit the decomposition. Are these points clear to all? Please let me know. Then talking about the energy flow, this is the third aspect of any ecosystem. See, sun is the only source of energy for all the ecosystems on earth except for deep sea hydrothermal ecosystem. For deep sea, it is the inherent energy of earth. Now, a second very important line here, less than 50% of the total energy of sun that reaches the earth, only 50% of it is photosynthetically active radiation. We call it PAR. See, the energy that reaches the earth is actually called total insolation, right? Now, out of the total energy reaching total insolation, only 50% is photosynthetically active radiation, that is PAR. Now, out of 50% power, plants capture only 2 to 10% of power. Another very important line. Are these points clear? Very, very important line. Now, there is always unidirectional flow of energy from sun to producers and then to consumers. This is the order of energy flow. Are these clear? Now, first law of thermodynamics is applied here. So we see all the three laws being practiced here. So first law says that there is unidirectional flow of energy. Now, second law of thermodynamics says that there is constant need to supply energy to synthesize the molecules so that we can counteract the universal tendency towards disorderliness. So yes, it follows the second law also. Are these points clear to everyone? Please let me know that you are understanding what I'm reading here, what NCERT has said. Yes, please. Then green plants are the producers. In terrestrial ecosystem, the major producers, see the examples of major producers in terrestrial ecosystem, it is herbaceous and woody plants. While in aquatic ecosystem, the producers are phytoplankton, algae, and higher plants. Are you noting now? Are you doing it 1, 2, 3? Are you doing this 1, 2, 3? This is 1, this is 2, this is 3. Please let me know. Okay, then coming on to the next important line, see here, death of organism is the beginning of detritus food chain. So we have basically two types of food chains running in the ecosystem. One is detritus and second is grazing food chain. Okay, now consumers are always heterotrophs. So primary consumers means who directly depend on the plants are the herbivores. The examples are insects, birds, mammals in terrestrial ecosystem and mollusks in the aquatic ecosystem. There are others also, but this is the definition, example given by NCERT. Now, what are primary carnivores? So primary carnivores are which eat herbivores. So they are basically, yeah. So primary carnivores are the secondary consumers. Is that clear? Now, what is a grazing food chain? Grazing food chain always starts with the producer like grass eaten by primary consumer, let's say a herbivore, for example, goat, and then goat is eaten by man who's a secondary consumer. While man can hold a different position also depending whether he's a vegetarian or a non-vegetarian. The second type of food chain is detritus food chain. Detritus means dead body, right? Detritus food chain always begins with the dead organic matter. So who's going to act on the dead organic matter? Decomposers. So detritus food chain always starts with detritus and it is made up of decomposers who are heterotrophs, who are saprotrophs. Now, one very, very important line here, please underline that in aquatic ecosystem, guys, GFC, that is grazing food chain. In aquatic ecosystem, grazing food chain, grazing food chain is the main conduit of energy flow. Very important. In aquatic, it is GFC, while in terrestrial ecosystem, large energy comes through detritus food chain, that is DFC. This is very, very important. You should write it separately here. In aquatic, it is the GFC, and in terrestrial, it is the DFC, detritus food chain. Is that point clear? So you can remember AGTD. So aquatic, GFC, and terrestrial, it is DFC. Please make sure that you do write down these points. Now, naturally, interconnected food chains, they form the food web. Is that clear? Then each trophic level has a certain mass of living material at a particular time. This is known as standing crop, again a PYQ. Now the standing crop is measured as a mass of living organism, which is called biomass, or the number in the unit area. Now measurement of biomass is always more accurate if we consider the dry weight. Why is it so? Because now there will be no change in the weight because the water is already evaporated. Is this point clear? Is this clear? Now, what will be the safe score for physiotherapy? Anurag, let's talk about physiotherapy in a while. So, first of all, I think we should all focus on clearing our NEET exam. Isn't that so? It depends. It always depends that what is the final score, what is the cutoff, so we can decide accordingly. All right, coming on to 10% law, guys. Only 10% of the energy is transferred to each trophic level from one trophic level to the another. Is that clear? So this is 10% law. Then finally we have ecological pyramids. What are ecological pyramids? So this is a way of representation. This is a way of representation in which the base of the pyramid always represents the producer or the first trophic level, and the apex always represents the tertiary or the top level. Is that clear? Now we have three types of ecological pyramids: pyramid of number, pyramid of biomass, and pyramid of energy. See, by there is no exception to this, that pyramid of energy is always upright. See this, the pyramid of energy is always upright. It can never be inverted or of any other shape. Why? Because energy, as we have seen, according to the 10% law, the upper trophic level will only receive 10% of the lower trophic level. So as we go upwards, the energy will always go on decreasing. Is that clear? Energy will always go on decreasing. But if we talk about the other ecological pyramids, see here, these are important. Pyramid of number in the grassland ecosystem. Now, what is the shape here? This shape is known as upright. So, pyramid of number in grassland is upright. Is it clear? Then coming on to pyramid of biomass in the terrestrial region. Again, you can say grassland. Again, the shape is upright. Why? Because the biomass of producers is more than the herbivore, then primary carnivore, and then top carnivore. Is this clear? Then coming on to a pyramid of biomass in aquatic ecosystem. Now see, in aquatic ecosystem, the producers are the, let's say, algae, and then we have consumers like fishes, small fishes, and then still large fishes. Yes or no? So what happens? The biomass will go on increasing. Now the number of algae will be more. If I talk about the pyramid of number in aquatic ecosystem, the shape would be upright. So maximum algae, smaller number of small fishes, and even lesser number of large fishes. But if we talk about the pyramid of biomass here, the shape will be inverted because the biomass of large fishes will be much more. Are these points clear to all? Then we have this ideal pyramid. Which is the ideal pyramid? Pyramid of energy, because it is always upright. Now, trophic levels represent functional level and not a species level. This is the important statement. In most ecosystems, yes, in most ecosystems, guys, all the pyramids of number, energy, and biomass are generally upright. So producers are obviously more in number than the herbivores. But there are some exceptions. For example, if I talk about a tree ecosystem, then in that case, tree is the producer, but there is one tree and there are so many insects, sorry, there are so many small birds, and on the body of every bird, there are so many insects. So, what is the shape of a tree ecosystem? So, one tree, many birds, and then so many insects. Tree, birds, and insects. This is when we consider the pyramid of number. But if I talk about the pyramid of biomass, then it will be different. The pyramid biomass will show like this. The biomass of tree will be much more than the biomass of birds, and then insects will be even lesser. So this is pyramid of biomass for tree ecosystem. Is that clear? So are you able to understand? Now, what are the limitations, guys? Limitations of ecological pyramid here, that does not take into account the same species which can belong to one or more trophic levels. Why? Because it only considers a simple food chain and does not accommodate a food web. And saprophytes are not given any place in the ecological pyramid. Are these points clear to everyone? Please let me know. Please let me know. Ecosystem is the easiest chapter. I hope it stays easy for everyone, but otherwise NTA is giving quite a lot of importance to this chapter. NTA is giving a lot of importance, guys, to ecology over time. All right, moving on to the next chapter. Moving on to the next chapter here. Wait. Okay, moving on to the next chapter, which is organisms and population. Yes, please. Moving on to this chapter, organisms and populations. See, we'll directly jump onto this chapter page where we are talking about population attributes. Now, I've written down which line has already come in the PYQ. See here. So, population has certain attributes. What are the attributes? Number one is the birth rate. See, first is the birth rate, second is the death rate, and third is the sex ratio. Now, this was 2020 question. Is that clear? Now, second, we have been asked to calculate the birth rate. What is the formula for birth rate? Formula for birth rate is, see, how do we calculate? So this was a formula based numerical asked in 2023 and also in 2022. See, the question is, if a pond has 20 lotus plants last year and through reproduction 8 new plants are added, what is the current population? It will become 28. Now what is the birth rate? So how do you calculate birth rate? Birth rate is number of births upon the original population. That's it. So it is new upon original. New upon original. This is the formula for birth rate. Are these points clear? Similarly, if you have to calculate the death rate, how will you calculate? So number of deaths upon the original population. The data can change, but the formula will be this. Are you getting that? Are you getting that? So this is a very important thing. The third characteristic of a population is a sex ratio. Then coming on to age pyramids. See the number of years and the number of times NTA is asked from this topic. So see here, this first graph represents the expanding population or the growing population, while this is the stable population and this is the declining population. So the question has already been asked from expanding and stable. NTA can repeat the same thing, or declining population can be a new field of interest here. Okay, now see here. So the base always represents the pre-reproductive stage. Then the middle of the pyramid shows the reproductive age group, and the top represents the post-reproductive. Now the question is, in 2023 and 2018, the question was statement-based question from this graph, that in growing population, the maximum population belongs to pre-reproductive, intermediate to reproductive, and minimum population of the post-reproductive age group. Is that true? Is that true? Right? While in case of stable population, reproductive age group and pre-reproductive age group is almost equal in number. Is that clear? While in declining, what will happen? The post-reproductive and the pre-reproductive, they are both less. They are both minimum, while the reproductives are maximum. The reproductive age group is maximum in number. Is this clear? Then moving on to next part. Moving on to the next part. See here, we are going to talk about natality. Natality is the birth rate, and we have just seen how to calculate that. Number of births during a given period. This was 2018. This was 2018. Then we have mortality. Mortality is the death rate. Number of deaths in the population. Immigration is the number of individuals of same species coming into the habitat. And then emigration is the number of individuals of the population who have left the habitat or gone elsewhere. Are these points clear? Now then, this figure is important. This figure is important. So population density is determined by birth rate. So birth rate and immigration adds to the population density, while death rate and emigration decreases the population density. Are these points clear? So this is the formula. So NT plus 1 is the new population density. It is a new population density. The formula would be NT plus B plus I, that is birth rate and immigration are going to add, while death rate and emigration are going to subtract. And this is the original. Are these points clear? So this was the question. This was a line from 2024 here, that population density will increase if the number of births and immigrations are more. See here, population will increase if B plus I is more than the D plus E. Are these points clear to all? Please let me know that these things are clear. Please let me know that these points are clear to all. Are these points clear to everyone? Now, moving on to the next part, guys, give me. All right. Coming back here. So another very important topic from this chapter is growth models. See here. So there are two types of growth models. Every year we are getting some of the other question from this topic. So 2023 asked us about exponential growth. What is exponential growth? When the population goes on just increasing. Why? Because here the resources and the habitat are unlimited. So when does exponential occur? Exponential growth occurs when there is unlimited resources. Is this clear? See this graph. So this graph was asked in 2024. So this represents the exponential, this represents the exponential, and this S shape represents the, this represents the second one, the logistic. This represents the logistic growth. Now, the question was asked from here. What's written in the description of the graph? See here. So what does A represent? A is when the responses are not limiting the growth. Why? Because we have unlimited resources. Is this point clear to all? Is this point clear to all? And then second, sigmoid curve. So exponential gives us J-shaped curve. Is that clear? And what is the equation? dn by dt is equal to Rn. dn by dt is equal to Rn. Is it clear? Where R is the intrinsic rate of natural increase. Are these points clear? Are these points clear to all? And while this second graph is the logistic growth, what does logistic mean? When the resources are limited. And what is the shape of the graph? It is S-shaped. It is S-shaped. And what is the formula here? The equation is dn by dt is equal to Rn and then k minus n upon k. And what is written here? When the resources are limited, we get a logistic plot. Please let me know that these points are clear. Please let me know that these points are clear to all. Now see here. So dn by dt is equal to Rn. This is for exponential. We can also write it nt is equal to n naught ERT. So what is E? This was asked in 2021. See the importance of all these points here. E is the base of the natural logarithm and the value is 2.7. Even these things are asked. So you can't ignore that. You can't ignore that. Is this point clear? Please let me know that these points are clear to everyone. So this was logistic growth model. And then, sorry, this was exponential and logistic growth model. And for logistics, see what does NCERT say? A population growing in a habitat with limited resources, this is 2023. And the whole paragraph came as such in 2024. Came in 2024. See, population growing in habitat with limited resources, and whenever, guys, whenever there is a S-shaped curve, you know that initial part is known as the lag phase, then comes the exponential phase or phase of acceleration, and then comes the phase of this is exponential, this is lag, and then we have the stationary phase or the plateau phase, or we can call it the phase of deceleration. Phase of deceleration. Are these points clear? So, and finally, when it becomes parallel, this is known as asymptote. This is known as asymptote. So what is asymptote? Asymptote is where, see the graph again. See, this is asymptote. When it becomes parallel to K. This is asymptote. This is asymptote. Asymptote, sorry. Are these points clear to it? Where K is equal to N, this is the asymptote. Points clear to everyone? Now, it was Verhulst and Pearl who gave us the logistic growth. So what is dn by dt is equal to Rn k minus n. Are these points clear to everyone? K minus n upon k. This is dn by dt. Please let me know. So see, if kn minus k k minus n upon k is equal to 0, when will it be 0? When n upon k is equal to, then n upon k will be 1. Yes or no? Yes or no? So this is 2016 question. This is 2016 question. And what is K? K is the carrying capacity. What does K represent? K represents the carrying capacity. Please let me know that these points are clear to you all. Yes, guys. Please let me know these points are clear to you all. Quickly. Please let me know these points are clear to you all. Okay. Moving ahead then. Moving ahead then. All right. All right. Sorry. Okay. Coming on to population interactions. Now, this table of population interaction is another very, very important aspect of the chapter. See, how many times are we getting questions from this? So, if we talk about mutualism, it is a positive, positive interaction. Competition is a negative, negative interaction. So, predation is positive, negative. Parasitism is positive, negative. Commensalism is positive and neutral, positive and zero. So, commensalism is negative and 0. So you got this exact table in 2023, 2022 and 21, then 2016. Are these points clear to everyone? Please let me know, guys. Moving ahead, then, sorry. So which of the following interaction is used in the medical science for the production of antibiotics? So see, antibiotics are produced by some organisms, right? And for example, penicillium is producing a penicillin which is destroying staphylococcus. So staphylococcus is being damaged here, destroyed here, but there is no effect on penicillium. Yes or no? So penicillium is showing, this penicillium is showing neutral effect, no effect on penicillium, while staphylococcus is getting affected negatively. So this interaction is being used in medical science for the production of antibiotics. Such a beautiful question it is in 2018. Yes or no? Then another example, 2022, we got this question that for this is about predation, that predators keep the prey population under control and maintain the ecological balance. This was 2022. Please let me know that these points are clear to everyone. Then guys, another thing about predators, they help in maintaining the species diversity in the community. Now example of an important predator here is disaster. It is a starfish in the American coast of Pacific Ocean. So disaster is an important predator here. Please let me know that these points are clear to everyone. Now another very important line which occurred in 2022 NEET exam, if predator is too efficient, it will overexploit the prey and the prey will become extinct, and following it the predator will also become extinct because there will be lack of food. So the predators are very prudent. Predators are very prudent. They never overexploit their prey. They only hunt for the sake of hunger and not for any other reason. Now there are other examples of predation. Other examples, for example, see, how do prey species protect themselves or evade the predation? Number one, they show camouflage, or they might have some poisonous secretion. For example, monarch butterfly is highly distasteful to the predator. Yes, similarly, why it is distasteful? Because in its larval stage it has eaten a caterpillar. Sorry, it has eaten a poisonous weed in the larval stage. It has eaten a poisonous weed in the larval stage, which makes it very distasteful for the predators. Are these points clear to everyone? Now, next example is that all herbivores are the predators. See, we are all predators. Even if we are eating vegetables, we are eating them, right? We are predators. Then nearly 25% of all insects are phytophagous. That means they feed on the plant sap. Are these points clear? Moving on. Now, calotropis, this weed calotropis is able to protect itself from being eaten or grazed upon because of the presence of some highly poisonous cardiac glycosides. So why don't animals graze on the calotropis? Because they produce highly poisonous cardiac glycosides. So they cause cardiac arrest in the organism. So this is known as this is an example of chemical defense. Are these points clear to all? Please let me know that these points are clear to all. Similarly, guys, we extract several chemicals from the plants like nicotine, caffeine. They are all produced as a method of defense against grazers and browsers. Next, coming on to competition. So, for example, South American lakes visiting the flamingos and resident fishes compete for their common food, which is zooplankton. This is known as interspecific competition. So whenever there is competition between different species for food or for habitat, it is called interspecific competition. And this was 2024 ka question. Are these points clear? Please let me know that these points are clear to all. Then what is the exact definition of competition? It was asked in 2024. See here, it is best defined as a process in which fitness of one species is measured in terms of the intrinsic rate of increase and is significantly lower in the presence of another. So, what is competition? When your efficiency decreases, when your fitness decreases in the presence of other, that is competition, right? Now, in 2024, again, there is a concept called interference competition. So, what is interference competition? It means the efficiency of one organism will decrease in the presence of another organism. For example, a Bington tortoise in Galapagos Islands became extinct in the presence of goats. So when goats were introduced within 10 years of their introduction, the tortoise became extinct. Why? Now, why did this actually happen? Because of the greater browsing efficiency of the goats. So goats will eat the whole food, the whole grass, and there was hardly anything left for the tortoise. Is this clear? So what happens in the presence of goats? Their efficiency is going to decrease. So this is called interference competition. Are these points clear? Then we have another concept called competition release. So competitive release means if you remove that species from the vicinity, then the number will be restored again. So what happened when they removed the goats from the Galapagos Islands? The number of organisms, the number of tortoise started restoring. Is this point clear? Is this point clear? Yes, I am trying to hydrate myself time to time. Now, coming on to see 2024 paper was quite and it was very much about this ecology. Even 2025, 2024, NTA is liking these three chapters quite a lot. NTA is liking these chapters. So it is the last year we had around 10 questions. We had 10 questions, guys, from ecology. Okay. 10 questions means 40 marks. Do you understand the importance of ecology here? Then Connell's elegant field experiment. What is Connell's elegant field experiment? It says it showed that on a rocky sea coast of Scotland, the larger and competitively superior barnacle, Balanus, dominates the intertidal area and excludes the smaller barnacle, that is Chathamellus, from that zone. So basically what happens? Connell observed that a superior barnacle known as Balanus dominated the area so much that it just led to extinction or exclusion of the inferior species, which is Chathamellus. Similarly, in general, herbivores and plants appear to be more adversely affected by competition than carnivores. Is this clear? This line was in 2024, 2023. So as such, the lines are important from this chapter. Then comes a very famous Gauss competitive exclusion principle. 2016, 24, 23. So 2016, right? So all these years we have been getting question from this principle. It states that two closely related species competing for the same resources cannot coexist. So when we are fighting for the same resource, we cannot coexist indefinitely. So there will be a day then when competitively inferior one will be eliminated eventually. Don't you feel that this is already happening to us? This Gauss was so true for us. Isn't that so? Is that so? Yes or no? So maybe, we might be excluded. But then came resource partitioning. So there was another scientist who said that it is not always true. The species facing the extinction might evolve mechanisms to promote coexistence rather than exclusion. But I don't think that NEET follows this rule of resource partitioning. So species which are facing competition will promote coexistence rather than exclusion by dividing their resources. By dividing their resources, this is known as resource partitioning. Is that clear? So, what is the example of resource partitioning? One example is that five closely related species of warblers living on the same tree. They were able to avoid the competition and coexist through difference in their through behavioral differences in their foraging activities. So the pattern of their foraging, that means collecting food, that was different. Please let me know that these points are clear to you all. Coming on to parasitism. The parasitic mode of life ensures the free lodging and minerals. Free lodging. Free lodging means free shelter, free food, that is, and in return causing diseases. This is known as parasitism. This is known as parasitism. Now, guys, parasites have also evolved according to the host. So they are also host specific, and both of them tend to co-evolve. Is that clear? And the host has also evolved special mechanism for rejecting and resisting the parasite, and the vice versa. Is this point clear? So parasites have adopted many adaptations, like loss of unnecessary sense organs, adhesive organs to cling to the body, loss of digestive system, high reproductive capacity, and the life cycle is often very complex. Means many hosts. It involves many hosts and many larval stages also. Is this point clear? Please let me know that these points are clear to you all. Please let me know. So many of the parasites harm the host, but they may reduce the survival, growth, and reproduction of the host and reduce its population density. Is this point clear? Please let me know. Please let me know. Now, parasites are of two types. We have ectoparasites and we also have endoparasites. Ectoparasites are which feed on the external surface of the host. Endo live inside the host body. Are these points clear? Ectoparasites are like lice, ticks on dogs, then many marine fishes, copypods on many marine fishes. Then fourth example is Cuscuta, right? So all these are different examples. Lice, ticks on dogs, copypods on the marine fishes, and Cuscuta, that is Amarbeg. Are these points clear? And then all these examples of liver fluke, tapeworm, which are so many in number, they are all endoparasites. They are living in liver, kidney. They may live in your lungs or RBCs. Is it clear? Or lymph vessels. All those which live inside the host body are endoparasites. Is this point clear to all? Then comes the brood parasitism. Brood parasitism. This is shown by a bird called cuckoo. Shown by a bird called cuckoo or quail. So what is brood parasitism? Parasitic bird lays its eggs in the nest of its host. Please let me know that these points are clear to you all. So cuckoo and the crow. So cuckoo is the parasite and crow is the host. Is it clear? We have another interaction, guys, known as commensalism. See, commensalism is a positive and neutral interaction where one is benefited while the other is neither benefited nor harmed. Okay. So one example is epiphyte on the mango branch. So epiphytes are the plants which grow on other trees. They just use their shelter, not and they don't acquire any food from the plant on which it is growing. So this is a epiphyte. Epiphyte on the mango branch. Similarly, second example is barnacles growing on the back of the whale. This is a second example. Third example is cattle ergot and the grazing cattle. So cattle ergot is a bird which keeps sitting on the grazing cattle and wait for grazing cattle to plow the soil so that they can find out insects from the plowed up soil. Is this point clear? Then, so ergots forage. See, this is the explanation. Ergots forage close to the cattle because cattle will stir up and flush out the insects, and ergots will be able to find and catch their prey. This was all in 2024. Are you able to see the importance of these chapters? Are you able to see? Now, very important example is fig flower and the wasp. So last year we got this question that fig flower is a non-vegetarian fruit. Is that clear? So first of all, VASP is not going to show this mutual relationship with the fruit. So VASP has this relationship with the fig flower and not the fruit. And secondly, we can't call it a non-vegetarian. So it is not the fruit, it is the flower with which it shows mutualism. See that? Is this clear? Then coming on to mutualism, yes. So it is a mutualistic behavior where both of them are getting benefited. For example, lichens. For example, mycorrhiza. Third example is between wasp and the fig flower and not the fig fruit. Are these points clear to all? Yes or no? Then fourth example is animals and the pollinating agents. Sorry, animals are the pollinating agents and the plants that they pollinate. This is the fourth example. This is also example of mutualism. So you are directly asked the examples in the form of match the following. Now see this paragraph. So fig species can be pollinated by the partner wasp fig species. So the female wasp uses the fruit not only as a oviposition or the egg laying, but also uses the developing seeds within the fruit for nourishing its larva. The wasp pollinates the fig inflorescence. So what is pollinated basically? The inflorescence. But later on, it will be using the fruit also. The first connection that it shows is with the flower, where it lays down its eggs safely, but later on when the larva comes out, see the adult will show that mutualism with the flower, but the larva is going to show mutualism with the fruit. So the female wasp uses the fruit not only as the oviposition, but also the seeds are going to be used by the larva. Please let me know that these points are clear. Please let me know that these points are clear. Then we have another example of Mediterranean orchid known as Ophrys, which employs the sexual deceit. Now, what do we understand by this? So, one of the petals of the flower resembles the female of the bee in size, color, and markings. So, male is going to be attracted to the flower, perceiving it to be the female, and it will pseudo copulate with the flower. Yes or no? So, this is how the flower gets pollinated. So, we see that the organisms which are codependent on each other, they always show co-evolution. They always show co-evolution and also the co-extinction. Now 2024 has asked the examples for mutualism. See, lichens, mycorrhiza, plant and the pollinator. What are the different examples of plant and the pollinator? Fig and the wasp, this has been asked. Orchid and the bee, which also exhibits sexual deceit, and then yucca and the plant. Please let me know that all these points are clear. All of you. So guys, we have completed ecology. Please let me know how many of you have joined me since the beginning and we have completed the ecology together. Let me know, all of you. Yes, please. Let me know that you have completed this ecology with me already. Yes, please. Let me know, guys, how many of you have completed ecology with me since beginning, or at least the 2 chapters, or at least the 2 chapters. Yes, please. Who has done only 1 chapter? Write down 3, 2, 1. All the 3 chapters or the 2 chapters or 1 chapter, let me know in the chat. And guys, now we are going to start with the next unit, which is biotechnology. We are going to start with the next unit. 3, 2, 1. Thank you, Jyoti. So we have already completed ecology, guys. We have already completed ecology, and we are going to start with biotechnology. All ready? All ready for biotechnology? Please let me know. All right then. Let's start the biotechnology unit and its biotechnology and its applications. So first of all here, the 3 critical areas of research in biotechnology are for providing the best catalyst in the form of improved organism using usually a microbe or a pure enzyme, then providing critical conditions through engineering for a catalyst, and third, downstreaming process technologies. These are the 3 critical areas of research in biotechnology. Please underline. Please underline. Ma'am, you are very fast. Yadla, then I can suggest you that you read it later on by decreasing the speed of the session. But I think this is what is needed at this point of hour. I think this is what is needed at this point of hour. How many of you agree? Tell me honestly, how many of you want me? Yes, please. How many of you want me to decrease my speed? Yes, please. Honestly, tell me. Write down slow. Write down slow. Those of you who want me to slow down, just write down slow in the chat. And then I'll tell you what it will bring up. What will the slow speed bring up? How many of you want me to go slow? Write down slow or fast. Only Yadla says slow. Vinu King says I need light speed. Fast speed. Gurnur says slow. Fast, slow. There are very few students who are saying slow. For those of you who want light speed, you can watch it at light speed later on by clicking on to 2.0 speed. And those who want it to be at a slower speed can reduce the speed as well. But I think if I slow down, guys, if you slow down at this hour, it can cost us a NEET rank. It can cost us revision. The revision that we can do at least twice more. Yes, it is going to take more time and it is going to cost us heavily. So that is why rather you try to increase your speed. See, I think it's slow. It is already slow. It is average. Yes, it is average, I feel. Honestly, when I was preparing these notes, I was going quite fast. Thank you so much. Okay, let's move on. So guys, majority wins the race. Okay, so majority of you are feeling that it is going at the right speed and you want a faster pace only. Then let's go with the majority. Okay, and those of you who are finding it difficult to cope up, my very honest suggestion would be you can listen to it later at a slower speed. Okay, let's go. Let's continue with the chapter of biotechnology. So, okay, listen. Now, biotechnology applications in agriculture. So, what are the applications in agriculture? First of all, agrochemical based agriculture. Second is organic agriculture. And third is genetically engineered crop based agriculture. So these are the three options that can be brought for increasing the food production. See, this is important. What all can we do to increase the food production? Three things to increase the food production. Wait, increasing the food production is either the agriculture can be agrochemical based, we can use lots of chemical fertilizers and pesticides and all that, or we can go for organic agriculture, or the third option for increasing the food production is going for or choosing the genetically engineered crops. See, green revolution was basically agrochemical based agriculture. Agrochemical based agriculture. So what happened in green revolution? So there was tripling of the food supply by using improved crop varieties, better management practices, and agrochemicals like fertilizers and pesticides. But the problem with green revolution was, what was the criticism of green revolution? That not all farmers uh, what do you say, they can't afford these expensive agrochemicals, right? Also, the varieties that were used, no doubt they showed increased yield, but this yield was not possible using the conventional breeding methods. Is that clear? And also, guys, the chemicals that were used during green revolution, they had lots and lots of side effects. Is this clear? Then comes this very important new topic, tissue culture. Tissue culture. Let's talk about this. What is tissue culture? So this was a technique for crop improvement, another technology for crop improvement, which was developed and especially during 1950s. So what do we do? So whole plant can be regenerated from the explant. So what is explant? We can use any part of the plant to develop a new plant. So part used to develop a new plant is known as an explant. Is it clear? So basically what do? We take an explant in a test tube, we give it sterile conditions, sterile conditions, and we give it nutrient medium, and also we add some hormones. Yes, we add some hormones to make it first a callus. So explant is used to make a callus. What is callus basically? So callus is a mass of cells which has the capacity to develop into anything. So basically callus is a group of meristematic cells. They are undifferentiated cells. Is that clear? Now this callus has the capacity to generate the whole plant from any into any other tissue, and this is known as totipotency. So the capacity to generate a whole plant from any cell or explant is known as totipotency. So basically, from a well differentiated tissue, we make it a callus first. Callus is undifferentiated. Is that clear? We make it undifferentiated. All right, just a second, guys. Is this much clear to all? So we use any part of the plant to grow a complete new plant. Okay, this is tissue culture. Then we have another thing called micropropagation. So what is micropropagation? So in micropropagation, the method of producing thousands of plants using the tissue culture. Since we are using microscopic structures to propagate the plant, we also call it micropropagation. Is it clear? Now, of course, the plants which will grow from the X plant, they will be identical to the original plant. Yes or no? So that is why all the plants which are formed from the X plant by this micropropagation or tissue culture, they are known as soma clones. They are known as soma clones. So plants like tomato, banana, apple, etc., they have been produced by the help of this tissue culture on commercial scale. Please let me know that these points are clear to you all. Please let me know. So the part of the plant which is actually used as an explant, the best part of the plant to be used as an explant is always meristem. Is always meristem. It can be apical or it can be axillary. Why? Because this part is free of virus. Is this clear? So we can always remove the meristem and grow it in vitro to obtain the virus-free plants. This was in 2014. This was in 2014. Is this point clear to all? Is this point clear to all? Then another process we have here is somatic hybridization. Another process we can use is somatic hybridization. So what do we do in somatic hybridization? So basically we isolate the two protoplasts from two different varieties of plants, and they are fused together. So when the protoplasms are fused, that resultant protoplast is known as hybrid protoplast. Is this clear? This was 2024. So hybrid protoplast is produced by fusing the two protoplasts, right? Now this hybrid protoplast is grown to form the new plant. So the hybrids formed here are known as the somatic hybrids, while the process is known as somatic hybridization. Are these points clear? Now what is the example of somatic hybridization? We have formed, see, formation of pomato. Now pomato is formed by the fusion of tomato and potato, and it has the characteristics of both. Is this clear? The plant did not have all the desired combinations, but so it was not used for commercial. We don't get it in the market, but at least it was an experiment worth doing. Is this clear? Now, then we have third. See, we have read that there are three different ways of enhancing the food production. One is agrochemical based, and we've talked about green revolution. Then we have talked about other two techniques, tissue culture or somatic hybridization. And third comes the genetically modified organisms. Now, what is the usefulness of GMOs? So first, crops can be made tolerant to the abiotic stresses. Second, their dependence on chemical pesticides can be reduced. So pest-resistant crops are produced. Then third, to reduce the post-harvest losses, increase the efficiency of mineral usage, and enhance the nutritional value of the food. Guys, this is known as biofortification. This is known as biofortification. Is this point clear? For example, golden rice. Now, golden rice is enriched with vitamin A. This is actually golden in color and it has more of vitamin A. So it is an example of GMO. It is an example of GMO for biofortification. Please let me know that these points are clear. Right? Then moving on to next, that is Bacillus thuringiensis. See, we are going to talk about the use of biotechnology in agriculture, right? We are going to talk about use of biotechnology in agriculture. So second example is usage of this bacteria Bacillus thuringiensis to form BT, that is BT cotton, BT corn, BT rice, BT potato or tomato and soybean. Now all these plants are known as biopesticides. Why? Because these plants will not be eaten by the pests. So basically Bacillus thuringiensis is an example of a biopesticide because it will not allow the pests to eat these crops. Are you clear? So what is pesticide? Which destroys the pest and protects the crop. But here we are using a living organism as a pesticide. So hence biopesticide. Is it clear? Which crops are the example in which we have used these biopesticide? BT cotton and all these. Are these points clear? So this was in 2020 and 2024. Now how is this BT cotton actually made? So first of all, you must know that Bacillus thuringiensis, this bacteria, it produces the protein. It produces the protein and which kills the insects of three different, see this, of three different uh, orders. So Lepidopterans, Dipterans and Coleopterans. So what are the examples of Lepidopterans? Tobacco budworm and emmyworm. Coleopterans, beetles, and Dipterans includes flies and mosquitoes. So basically BT cotton, BT, that is Bacillus thuringiensis, it forms crystal proteins. Is that clear? And this protein is insecticidal in nature. Now, why does this toxin not kill the bacillus? Because it is produced in the inactive form. It is produced in the inactive form. Now, this whole paragraph is so important. See, you've gotten so many questions from this paragraph. So many questions from this paragraph. Is it clear? So the activated toxin binds to the surface of the midgut epithelial cells, creates spores and cause cell lysis and swellings and eventually the death of the insect. So this whole paragraph came in NEET as a sequence based question. 2024, they asked the process of BT in which how it protects the crop. So BT toxin kills by creating the pores. This was 2024. Is this point clear to all? Is this point clear to all? Please let me know. Then moving on to BT toxins are always insect group specific. For example, see what is the name of the toxin? BT toxin is cry. BT toxin is capital CY. This is the BT toxin. Toxin is basically a protein. So it is a cry protein. Now this cry protein is produced by a gene. The name of gene starts with small c. Is that clear? So the name of the protein starts with small c. Wait. Alright, let it go. So BT toxins are insect group specific. There are different types of genes and these genes will be specific for different types of insects. For example, Cry1AC and Cry2AB, it controls the cotton bollworms and Cry1AB controls the corn borer. You have already gotten this question in match the following MTF. Is this points clear? Is it clear to everyone? Please let me know. Then talking about another application of biotechnology in the field of agriculture, that is producing pest-free plants. Second example is of a tobacco plant. See, there is a nematode in the name of Meloidium incognitia, which infects the roots of tobacco plant. So which part of tobacco is infected? Roots. Name of the roundworm is Meloidium. Is that clear? Now the strategy used here is RNA interference. It is RNA interference. Is this clear? So basically this technique is already present in all the eukaryotic organisms as a method of cellular defense. It is a method of cellular defense in all the eukaryotes. And this is how all the eukaryotes protect themselves from the viral infections also. So basically this How does this technique work? This technique involves silencing of specific mRNA or the complementary that mRNA molecule that binds to and prevents the translation of mRNA. This is known as silencing. These lines are all very, very important. Very, very important. Are these points clear? Okay. Now, using the agrobacterium vectors, how do we inject, how do we insert the foreign gene? Using a vector, and the vector used here is agrobacterium. So, using agrobacterium vector, nematode specific genes have been introduced into the host plant, which is a host plant, a tobacco plant. Are these points clear? So, it involves the production of both sense and antisense RNA in the host cell. The two RNAs being complementary, they will form double-stranded RNA and initiates, and this is how RNA will be silenced. This is how RNA will be silenced. See, parasite could not survive in the transgenic host expressing the specific interfering RNA. Is this point clear to all? All these lines are important, guys. All these lines are important. Now, coming on to the application of biotechnology in the field of medicine. Now, first, human hormone produced by rDNA technology or biotechnology was genetically engineered insulin. Genetically engineered insulin, and which host was used? Using E. coli as the host. Using E. coli as the host. Are these points clear? So, insulin is used for diabetes. The insulin which was used for diabetic patients was earlier extracted from cattle and pigs, slaughtered cattle and pigs, and this insulin was known as pigulin. But what was the problem with this pigulin? It developed allergies. It caused allergies. So reactions to the foreign protein took place. Is that clear? But later on, we tried to understand the actual structure of insulin. See here, the actual structure of insulin here. Yes, guys, let's understand the actual structure of insulin. Insulin actually has two short polypeptide chains. How many chains are present in insulin? Insulin has two polypeptide chains, two PP chains. One is A and second is B. Second is B. And both of them, they are joined, they are linked together by disulfide bonds, SS bonds. Are these clear? Then, when we understood this structure, what happened in mammals? This insulin is not produced directly. Rather, insulin is synthesized in the form of proinsulin. It is formed in the form of proinsulin like a proenzyme. Is it clear? And later on, it is fully matured and made functional to contain an C. It contains an extra stretch, that is a C peptide. The insulin contains two polypeptides, but proinsulin has three. It has three. Can you see it here? A, B, and C. This was asked in 2021. So mature insulin will have only two polypeptides. Three polypeptide chains, two polypeptide chains. So basically, we are going to remove this C chain. See the structure here. So A chain, B chain, and in between A and B, we have this C chain. So when we remove this C chain, we get a mature insulin. This is insulin and this is proinsulin. Now, it was Eli Lilly. It was Eli Lilly in 1983, the first American company which was able to first produce this successfully. Now, prepare two different DNA sequences corresponding to A and B chains and introduce them in the plasmids of E. coli to produce the insulin chains separately. See, chains of A and B were produced separately, extracted, and then combined by producing disulfide bonds. Now, this paragraph is super, super, super important. Please give me a thumbs up that you are all clear with this paragraph, and just see the importance. Just see how many times we got questions from this topic. Give me a thumbs up. Yes, guys. Anji. Let me know, guys. Let me know. Please let me know that all these points are clear. All right. Moving on. to the next topic here is gene therapy. Next topic is gene therapy. So the first case of gene therapy was in 1990. Okay, so what is gene therapy here? It allows the correction of gene defect. The genes are inserted in the person's cells and tissues to treat the disease. The first clinical case of gene therapy was in 1990 in a 4-year-old girl with ADA deficiency, and that is adenosine deaminase. Now, this enzyme is important for the immune system. So, in ADA deficiency, what happens? Your immune system doesn't develop. It leads to a disease known as SCID. So, this deficiency was corrected by, it was treated by enzyme replacement therapy, first of all. So, we were providing that enzyme from the external source. But what happens? The functional ADA, the final treatment was introduction of ADA complementary DNA introduced in the lymphocytes. Is that clear? So the whole point is that we should add this DNA, add this gene into the lymphocyte itself. But subsequently, what will happen? The lymphocytes will also die after a week or so. So this was also not a foolproof treatment. So first treatment was enzyme replacement therapy. Second method which was employed was adding a functional ADA or a enzyme in the lymphocyte. Is that clear? ADA producing ADA producing gene into the lymphocyte. But lymphocytes are going to die because, see, clearly written, these cells are not immortal, means they will also die. So the patient requires a periodic infusion of such genetically engineered lymphocytes. So these both were not the final treatments. The final treatment would be, see here, to isolate this gene from the marrow cells and which produce ADA and put it in the early embryonic stage. Now that would be the permanent cure. That would be the permanent cure. These two are just the treatments, but not a final cure. Not final cure. Are these points clear to all? Embryonic stage, it will be curable. It can only be cured during the embryonic stage. Now, another application in the field of medicine is through molecular diagnosis, guys. So, conventional methods of diagnosis include serum or urine analysis. But the problem with the serum and urine analysis is it does not serve purpose for early diagnosis of the disease. Okay. So, by the time symptoms appear, only then we go for serum and urine analysis. But by that time, the disease has already occurred. The disease has already occurred and the tissue damage has already taken place. So molecular diagnosis comes in by giving us some methods of finding the disease in the asymptomatic stage. So this is the benefit of molecular diagnosis. So there are different methods. One is recombinant rDNA technology. Yes. Then we have PCR, and third is ELISA, and the fourth is autoradiography. See here. Fourth would be autoradiography. Here. This autoradiography. Is it clear? There are 4 methods of molecular diagnosis. What is ELISA? It is a diagnostic. This is the diagnostic test which we use for AIDS, right? Then, what is autoradiography? So, in autoradiography, single-stranded DNA or RNA tagged with a radioactive molecule is used to hybridize the complementary DNA in a clone. Used for detection of mutated gene. So we use autoradiography to find out if the gene is mutated or not. Is this point clear? Then in case of ELISA, the principle on which it is based is antibody antigen reaction. And then we have applications of biotechnology in the field of animals. This is to study the normal physiology and development. So we can study the effect of complex factors in growth like insulin growth factors by introducing the genes and altering the formation of the factor. Is that clear? Second is to study the disease. The diseases which have been studied using the transgenic models are cancer, cystic fibrosis, rheumatoid arthritis, and Alzheimer's. Is that clear? Then biological products which have been produced. The biological products which have been produced using biotechnology. Number one is alpha 1 antitrypsin, which is used for the treatment of emphysema. Second example is production of human alpha lactalbumin in a cow known as Rosie. So basically, this Rosie has a gene, a human gene, okay, which will produce a human protein, that is human alpha lactalbumin. So human protein enriched milk is produced by Rosie, and how much protein is present in its milk? 2.4 gram per liter. So this milk is nutritionally more balanced product for human babies than the natural cow's milk. Is this clear? Then we can study the vaccine safety by studying it on the transgenic mice. Is it clear? Before giving it to the humans. And then chemical safety testing can also be done by producing transgenic animals which carry the gene and make them more sensitive to the toxic substance than the non-transgenic animals. So this is, then it is exposed to the toxic substance and the effects are studied. Please let me know that these points are clear to you all. Then there are some ethical issues also related to biotechnology. So some ethical standards are required to evaluate the morality. Is that clear? Some ethical standards are required that might help activities of all the humans, that might help or harm the living organism. So there has to be a check whether the research which is being done is harmful or helpful for the humanity. So somebody has to keep a check. Now see, therefore, Indian government has set up an organization in the name of GEAC, which is Genetic Engineering Approval Committee. So what does it do? It takes decisions regarding the validity of GM research and safety of introducing GM organisms for public services. Are these points clear to you? Are these points clear to you? Please let me know. Then there are some stories of some, which involved biopiracy, which involved biopiracy. See, what is biopiracy here? Yes, please. So what happened? So American company got patents on basmati rice through US patent, and they allowed the company to sell new variety of basmati in US and abroad. So this new variety of basmati was basically derived from Indian farmers variety. Is that clear? So this was one case of biopiracy. Second case, so what is biopiracy basically? So use of bioresources by the multinational companies and other organizations without proper authorization from the country and people concerned, and without giving the compensatory payment, without giving a compensatory payment. Is that clear? So this is an example of biopiracy. Definition of biopiracy is clear, and this is the example of biopiracy. Is this point clear to all? Yes or no? So similar, many such cases have already taken place, but India managed to win the cases. Then is this point clear to all? And then guys, Indian Parliament has recently cleared the second amendment of Indian Patents Bill to take into consideration the patent terms of emergency provisions, research and development initiatives. Please let me know that these points are clear to you all. And with this, the applications is done. And now moving on to the next chapter of biotechnology, which is principles and processes. Yes, guys. Starting with the principles. So the two major core techniques of biotechnology are genetic engineering. Yes, please. The two core techniques are genetic engineering and second is bioprocess engineering. Second is bioprocess engineering. Clear? What is genetic engineering? It is technique to alter the chemistry of DNA or RNA, while bioprocess engineering is maintenance of sterile or contamination-free ambience, so that the growth of only desired microbe or cell can take place, and the manufacture of biotechnological products like these can also take place. Please let me know that these points are clear. Then guys, the techniques of genetic engineering, what are the different techniques involved? It is rDNA technology, gene cloning and gene transfer. Is this clear to all? Now what is See, this line is very important. It was in 2022 that cloning or making multiple identical copies of any template DNA, the alien DNA is going to be linked with this part. In order to make a clone, an alien DNA is linked to a special part which is known as origin of replication. So any alien DNA can replicate and multiply itself in the host cell and can form multiple identical copies of the template DNA. This is known as cloning. This is known as cloning. Moving now to the next part, which all substances can be used. See, so we can use a native plasmid. So cutting of DNA at the specific locations became possible with the discovery of so-called molecular scissors. So molecular scissors are the restriction enzymes. These are the restriction enzymes and they belong to the class of endonucleases. This was 2023. And they are linked with the cut piece of DNA was linked with the plasmid DNA. Then coming on to tools of rDNA technology. See, what are the tools? Restriction enzymes, polymerase enzymes, ligases, vectors, and the host. So this was 2025. So the question was, which of the following are not used during the rDNA technology? So guys, we don't use DNA mutase or DNA recombinase. We don't use these enzymes. This was 2025. Then Hind2 always cuts the DNA at a particular point and that point is known as a recognition site. And the recognition site of Hind2 consists of 6 base pairs. Hind2 was the first restriction enzyme to be discovered, not isolated. It was first discovered. Is that clear? Then this line that today we have more than 900 restriction enzymes isolated from around 230 strains of bacteria. Now how do we name them? See, in this name of a restriction enzyme, the first letter, for example, this eco, this comes from the genus and the species. E stands for Escherichia and CO stands for the species, E. coli. Is that clear? R stands for, R is the strain, the rough strain. And this number is the order of isolation, not discovery. It is the order of, indicate the order in which the enzymes are isolated. Are these points clear? So, these restriction enzymes belong to a class of nucleases which are of two types, exo and endo. Exo will cut it from the ends of the DNA. Endo is going to cut in between at specific locations within the DNA. Now, each restriction enzyme functions by inspecting the length of a DNA sequence. Very important line. It finds its specific recognition site, will bind to the DNA, cut each of the two strands of the double helix at a specific point in the sugar phosphate backbone. So it recognizes a specific palindromic sequence. What is palindrome? Which reads the same forward and backward. Then this figure, very important. So E. coli cuts the DNA between the bases G and A. And only when the sequence GAATTC is present in the DNA. Is that clear? So when E. coli cuts DNA, it forms sticky ends. Are these points clear? Then see, the palindromic sequence of E. coli 1 is GAATTC. And see the polarity 5 to 3 dash. And similarly, so gut tuck we call it. This goes from 5 to 3 dash. And similarly, on the opposite strand, it is GAATTC. Is this clear? Then restriction enzymes for blunt ends. They may form the blunt ends also. Blunt ends is when it cuts right in the center of the palindrome site. Is that clear? Then overhanging stretches are known as sticky ends. Now the stickiness of the ends facilitate the action of DNA ligase. This is where the two DNAs are going to bind to each other. They are used in genetic engineering. Again, this figure is very, very important. Same restriction enzyme cutting both the foreign DNA and the vector DNA. So this is important. whether different enzyme is used or the same enzyme is used for cutting the two DNAs. It is the same enzyme. See here, it is the same enzyme. Clear? So there was this statement in 2019 that enzyme binds DNA to specific site and cuts only one of the two strands. No, it cuts both the strands. This is wrong. It cuts both the strands at both the strands, see, with the same restriction enzyme. Is this clear? Then ligase will join the foreign DNA to the plasmid and joins the two DNA molecules. Any doubt regarding this? Then guys, this topic is also, this whole page is also very, very important. See, so the fragments of DNA can be separated using the technique known as gel electrophoresis. Now the DNA fragments are always negatively charged. They are always negatively charged. So being negative, they will move towards the positive and they move towards the anode. They move towards the anode. Now the gel that we use in electrophoresis is agarose gel, which is a natural polymer obtained from seaweeds. Gelidium, Dracularia. I hope you remember. These are red algae. Then the DNA fragments separate according to their size through the sieving effect through the gel. The smaller the fragment size, farther it moves. So which one is going to move the quickest towards the anode? It is the smallest one. Is that clear? The longer one will be towards the cathode. Clear? Now the separated DNA fragments can be visualized not directly but by staining them with ethidium bromide. By staining them with ethidium bromide. So ethidium bromide is used, and then we don't use a white light for this. We use UV radiation to observe these bands. So bright colored, orange colored bands are visible under UV light of ethidium bromide. Please let me know that these points are clear. Now the separated bands of DNA are cut out from this, and this last step is known as elution. The fragments are purified by using the DNA fragments purified in this way are used in constructing the rDNA. Are these points clear? So DNA moves from negative to positive because itself is negative. Then examples. Commonly used cloning factors in humans are BAC, YAC, PBR322. This was 2014 question. Also asked in 2023. Then other examples of cloning vectors are plasmids, bacteriophages, that is the virus. So vectors are used for linking of foreign DNA for cloning. Please let me know that all these points are clear to all. So what are the features of a cloning vector? So cloning vector should have these three qualities. One, it should have ori, it should have a selectable marker, and it should have one cloning site preferably. Is this clear? Now let's read. It says ori is the sequence from where replication starts, also responsible for controlling the copy number. Then selectable marker. The vector requires a selectable marker which helps it to identify the transformant from the non-transformant. Is it clear? To check for transformation. So it is a process through which a piece of DNA, transformation is a procedure through which a piece of DNA is introduced into the host cell, and subsequent study will be made. So genes encoding the resistance to antibiotics like ampicillin, all these antibiotics, chloramphenicol, tetracycline, kanamycin, they are all called selectable markers. Is that clear? So the normal E. coli will not carry the does not have resistance against these antibodies. So all those who have resistance are the transformants. Are these points clear? Then cloning sites. Preferably single cloning site should be present. Why? Otherwise that endonuclease restriction enzyme is going to cut it at multiple sites. So what do? We ligate the foreign gene at BAM1 site of tetracycline resistance, and it will lose the tetracycline resistance. Now see this figure, very, very important figure. So this is the diagram of a PBR322. This is a artificial plasmid that has been produced. Now in this we see two selectable markers. One is tetracycline resistance gene, and then we have AMP resistance gene. Now, the two sites at which the foreign gene can be introduced in tetracycline, the two sites are BAM-H1 and SAL1. Basically, these are the endonucleases. BAM-H1 can also act and SAL1 can also act, but at different locations. So, similarly, foreign gene can be introduced in ampicillin resistance using another two restriction enzymes, PVU1 and PST1. So, 2021 said if PST1 is used to insert a gene in ampicillin, then ampicillin resistance is conferred or not conferred. So if PST1 is used to insert a gene in EMP gene, then ampicillin resistance is not conferred. So resistance will, if we are inserting a foreign gene, then ampicillin resistance will be gone. It will not be conferred. Is that clear? So you must know which are the two sites, or you can say which are the two endonucleases in ampicillin resistance gene and in tetracycline resistance gene. Are these points clear to everyone? Now, E. coli, see, also be careful of what is written below the diagram. E. coli vector, PBR322. What is ROP? ROP codes for the proteins involved in the replication of the plasmid. So this was 2024. Please let me know all these points are clear. So pathogenic bacteria gains resistance to antibiotics due to changes in their plasmid. Is it clear? Because we have already read in bacteria also that plasmid is so important. What is importance? It gives antibiotic resistance apart from some other functions. Then alternative selectable markers have been developed which differentiate the recombinants from the non-recombinants. Is it clear? On the basis of the ability to produce color in the presence of chromogenic substance. Now here the enzyme we are using is beta galactosidase. So the selectable marker would be the gene for enzyme. This is the selectable marker in this case. So we can use many selectable markers. Is this clear? So the one in which the insertional inactivation has been done, so the chromogenic substance will not be produced. So basically blue colonies are basically which don't have an insert. So blue is a non-recombinant or non-transformant, and white ones are the recombinants. Remember this conclusion. Are these points clear to all? Then vectors for cloning genes in plants and animals. See, in order to deliver genes to transform the eukaryotic cell, we do it with the help of different vectors. For example, for plants, we use Agrobacterium tumefaciens to deliver a gene of piece of DNA known as tDNA to transform the normal plant cell into the tumor cell. Is this clear? Then for animals guys, it is retrovirus to transform the normal cell into the cancerous cell. Then we have tumor inducing. Why do we use Agrobacterium? Because Agrobacterium has this TI plasmid, tumor inducing, in Agrobacterium, which can easily modify the cloning vector, and which is modified. It is not made, it is no more pathogenic to the plants. Before using it as a vector, it is made harmless. Is this clear? It is modified. Then moving on to the next method, which is heat shock method. It is heat shock method. So what do we do in heat shock method? By choosing the host. So sudden heat shock is given, which is around 42 degrees Celsius. So we give it a quick heat shock that is around 42 degrees Celsius and put it back on ice. So basically this heat shock is going to open the pores and it enables the bacteria to take up the recombinant DNA. Another method is microinjection. So this is used for introducing alien DNA into basically an animal cell. Microinjection. Clear? Then we have gene gun or biolistics. These are actually used for plants, particularly more suitable for plants. So here cells are bombarded with high velocity of microparticles made up of gold and tungsten coated with the DNA. And the last method is the disarmed pathogen vectors. Are these points clear? Are these points clear to all? Now see the process. So basically the question comes from the sequence. So first step of recombinant DNA sequence, DNA technology is isolation of DNA by using these enzymes. Is it clear? And purified DNA is separated out or precipitated by using chilled ethanol. Then second step is cutting the DNA at specific locations, and we know how do we do it? By using restriction enzymes. Yes. Third, that DNA needs to be amplified. The one which is separated needs to be amplified using the process called PCR, using the process PCR, which is polymerase chain reaction. What are the different steps in PCR? The sequence is denaturation, annealing, and extension. So the sequence itself is important. So in denaturation, we separate the two strands of DNA by heating it at around 94 degrees Celsius, right? Then we add a primer to it. This is known as annealing and it occurs at 54 degrees Celsius. And then finally, the Taq polymerase comes in, which helps the polymerization of the complementary deoxyribonucleotides and leads to extension, which takes place at 72 degrees Celsius. So if we want to count how many new DNA particles will be produced, so the number formula is 2 raised to power n, while n is the number of cycles. Are these points clear to all? Are these points clear to all? Yes or no? So this is PCR. And during PCR, the enzyme that we are using during elongation is Taq polymerase, which is obtained from a thermostable bacteria. Which is thermostable, which is obtained from a thermophilic bacteria known as Thermus aquaticus. Yes, thermophilic bacteria, that is Thermus aquaticus. The next step of biotechnology process is insertion of our DNA into the host cell. So how do we do that? We have different methods. Just talked about it. Heat shock method, gene gun method, micro injection, and so on. Is that clear? Then finally, we are going to obtain the foreign gene product. Foreign gene product. Is this clear? So any protein encoding the gene is expressed in the heterologous host. It is known as the recombinant protein. So the protein which is produced in a different host who is not actually supposed to produce it is known as a recombinant gene. Is this clear? Now the large vessels which are used for producing them in a huge amount in the large quantity, those vessels are known as bioreactors. They are so large that they have the capacity of around 100 to 1000 liters. So it provides optimal conditions for achieving the desired product by providing optimal growth conditions of temperature, pH, substrate, salts, vitamins, and oxygen. Clear? Now the most commonly used bioreactor is of the stirring type, and this is the picture. So stirring type, see the labeling in top. At the top we have a motor, then we have a foam breaker, flat blade impeller, and the sterile air is added in. And see, in stir tank bioreactor, it facilitates the oxygen availability throughout the bioreactor. Now, bioreactor also has an agitator system, an oxygen delivery system, and a foam control system. Is that clear? Apart from that, it has a temperature control system, pH control system, and a sampling port from where we take out the sample for testing. Is this point clear? Now, last step of biotechnology is downstreaming process. The three steps in downstreaming are separation, purification, followed by preservation. And then it is to marketed for the usage. Is this point clear? Please let me know that this chapter is done. Guys, we are done with the second unit also. We are done with the biotechnology unit also. Everybody give me a thumbs up that we are thorough with this unit. Yes, please. How many of you have completed 2 units with me? How many of you have completed the 2 units with me? Yes, please. Quickly let me know. How many of you have completed the 2 units with me? You will get these PDFs, but first of all, you let me know. How many of you have completed? Give me a thumbs up that how many of you have completed 2 units with me. Yes, guys. All right, perfect, perfect. Let's move on to the next unit then. And the chapter is Microbes in Human Welfare, coming directly to, yes, please, to this page. So, microbes in household products. The first microbe that we talk about is lactobacillus. First product is the lactobacillus, lactic acid bacteria, which is used for making curd, right? And what does it do? It coagulates and partially digests the milk proteins. So converting milk to curd improves the nutritional quality by increasing B12. That is curd. That is why curd is better than milk. Is it clear? Second function of lab is it helps in checking the disease causing microbes. Second is for the formation of dough, which is for dosa and idli. We also use bacteria for the fermentation. Are these points clear? Then, in case of making bread, for making the dough for bread, we don't use bacteria, we use a yeast, which is Saccharomyces cerevisiae. Is that clear? Then we have this very important word toddy, a traditional drink of South India used by fermenting sap of palms. Then cheese was one of the oldest food. Then there are different types of cheese based on the characteristic flavor, taste, and texture. Now, for example, Swiss cheese produced by Propionibacterium shermani, it has big holes. So large holes are because of large amount of CO2 produced. Then we have Roquefort cheese produced by a fungi. See, this Propionibacterium is a bacteria. Let's make it a little big. Yes, Propionibacterium is a bacteria, while Roquefort cheese requires a specific fungi. Are these points clear? Then moving on to microbes in industrial products. These microbes are grown in large vessels known as fermenters. Like we have bioreactors, we have fermenters. So the fermented beverages produced is like alcohol. So alcohol production, again by the yeast, by the same name, Saccharomyces cerevisiae, which is used for bread making. So we call it baker's yeast, but now it is also known as brewer's yeast. Is this clear? Then there are two types of alcohols produced based on the amount of alcohol in it. So without distillation, we have wine and beer. And then whiskey, brandy, and rum are produced by distillation. Are these points clear? Very, very important. Then moving on to antibiotics. So what are antibiotics? They're chemicals produced by some microbe and kills the growth of other organisms. For example, penicillium. Okay, so first antibiotic was penicillin, produced by Penicillium notatum, right? It is an effective antibiotic produced by Alexander Fleming while working on Staphylococcus bacteria. But the full potential was discovered by Ernest Chain and Howard Florey. They used it to treat the American soldiers during World War II, and therefore all three of them deserved a Nobel Prize. Then these antibiotics can only be used for bacterial diseases, not for viral. So they have capacity to improve the deadly diseases like plague, whooping cough, diphtheria, and leprosy. Is this point clear? Then chemicals, enzymes, and other bioactive molecules. These lines are all very, very important. See, Aspergillus niger, a fungus for citric acid, Acetobacter, a bacteria produces acetic acid. All match the following. All match the following. Then Clostridium botulicum, a bacteria producing butyric acid, lactobacillus, lactic acid. Are these clear? Then Saccharomyces cerevisiae is known to produce ethanol, the alcohol. Streptokinase produced by bacteria Streptococcus. It is a clot buster. Streptokinase is a clot buster. It is used for patients who have undergone myocardial infarction, which led to heart attack. So it is a first aid for heart attack patients. Then we have cyclosporin A, which is an immunosuppressive agent produced by a fungus, that is Trichoderma polysporum. Then we have statins. Statins are produced by again a fungus, Monascus purpureus. It is a blood cholesterol lowering agent. How does it work? Competitively inhibits the enzyme which is needed for synthesis of cholesterol. Is this point clear? Then coming on to Sewage treatment, very important. So primary treatment, guys, what happens? See, sewage treatment involves two major treatments, primary and secondary. Although there is tertiary, but NCERT doesn't talk about it, so we'll restrict to primary and secondary. So what happens in primary treatment? There is physical removal of particles. So all the solids will settle down in the form of primary sludge, and which will be removed, and then the supernatant is known as the effluent, and this effluent is sent for the biological treatment or the secondary treatment. So first of all, it is sent to the large aeration tanks where air is pumped into it. This air leads to the growth of useful aerobic microbes, bacteria and fungus, and form flocks. Right? Now flocks are going to degrade the organic matter. Is it clear? So these flocks are going to degrade the organic matter, which is going to reduce the BOD, biochemical oxygen demand. Now see, what is BOD? So basically BOD, see, BOD is the amount of oxygen needed that would be consumed in all the organic matter in 1 liter of water. It is needed by the bacteria to decompose the organic matter. So more the organic matter, it will demand more oxygen. Therefore BOD tests the rate of uptake of oxygen. It is indirectly the measure of organic matter. So greater the BOD, more is the pollution. Points clear to all? Yes. Coming on to activated sludge. See, once the BOD is reduced, organic matter is digested, we need to remove this flock also. So now it is sent to the settling tank. It is sent for the settling tank where we remove, where the bacterial flock is allowed to sediment. So for removal. So this is known as activated sludge. So what does activated sludge consist of? Bacterial flocks. Right? Now part of activated sludge is sent to the aeration tank, which will enhance the growth of flocks, and the remaining part is sent to the anaerobic sludge digesters. Now these anaerobic bacteria are going to degrade the flocks, and they might produce a mixture of gases like methane, H2S, and CO2. And this is a combination of biogas as well. Is this clear? So the effluent from the secondary treatment is generally released into the water bodies. Please let me know that all these points are clear. Yes, please. Also, guys, this was initiated by which ministry? Ministry of Environment and Forests, Ganga Action Plan and Yamuna Action Plan to save these major rivers. It was decided to to install STPs at the banks of all these major rivers. Yes, please. Are these points clear? Then production of biogas. So we know methane is the main gas in biogas along with CO2 and H2, produced by a special bacteria called methanogens, for example, methanobacterium. Is it clear? These same bacteria, we have also talked about them in arc bacteria. Yes, they are the arc bacteria found in the rumen of the cattle. Yes or no? And they cause breakdown of cellulose in their lumen. Please let me know that all these points clear. Now, since biogas production requires dung of the cattle, we also call this gas as gobar gas. We also call it a gobar gas. Now, there are special plants set up, biogas plants, with concrete tank of 10 to 15 feet deep. See this, see the labeling here. So here we add dung and water, and then this is where the bacteria are present called the digester, and biogas is produced. As it is produced, this gas holder will keep on rising. And once the dung is digested, we call it sludge, and it is taken out to be used as manure. Are these points clear to everyone? So they are built in the rural areas where there is surplus of dung. Is that clear? Now, this biogas is used for cooking and lighting. Now, the technology of biogas production was developed in India by two bodies, IRI and KVIC. What is Indian Agricultural Research Institute, and KVIC is Khadi and Village Industries Commission. Please let me know that these points are clear to all. Yes, please. Quickly let me know. And then, guys, we have biocontrol agents. These biocontrol agents, yes, what are biocontrol agents? Where we use biological methods for controlling the diseases instead of using the chemicals. Clear? Then There are some examples of biochemical agents, sorry, biocontrol agents. The first example is a ladybird and dragonfly. These are used to control aphids and mosquitoes. So this was in 2024. You can get match the following. Then we have already discussed Bacillus thuringiensis. These are all biocontrol agents. All the 5 examples are given here. Then next example is of Trichoderma. It is a free species of fungi that lives in the roots ecosystem and protects the plants from various diseases. And fifth one is Baculovirus. Belongs to the genus Nucleopolyhedrovirus. Excellent candidate for species-specific narrow spectrum insecticidal application. No side effects to any other group, and it is used in integrated pest management program and ecologically sensitive areas. Please let me know that these points are clear. Then we have examples of biofertilizers. In biofertilizers, guys, we have example of rhizobium, which is symbiotic and lives in the roots of leguminous plants. Now this is a bacteria. So the main sources of biofertilizers are bacteria, fungi, and cyanobacteria. So this is a symbiotic one, while we also have some free living like azospirillum and azotobacter. Then talking about fungi. So second is fungi. So in fungi, uh, we have a genus Glomus, which is generally involved in forming a symbiotic association with the roots of the plants. Now fungi and roots are known as mycorrhiza. What role does it play? Major role is in absorption of phosphorus from the soil and passes it to the plant. So also provides resistance to the root-borne diseases and tolerance to salinity and drought and overall increase in plant growth. Then coming on to third source of biofertilizer, which is cyanobacteria. So cyanobacteria we've read already in biological classification. It is autotrophic, and some of them can fix atmospheric nitrogen, like Enabina and Nostoc, because we have seen that they both contain heterocyst. Yes, but oscillatoria actually doesn't really fix nitrogen. Now, are these points clear? So, Enabina and Nostoc, the colonial forms, the filamentous forms, they have heterocyst. These are the special cells which fix nitrogen fixing cells. Are these points clear? But see, in paddy fields, cyanobacteria plays an important role as a biofertilizer. Please let me know that all these points are clear. And guys, with this, the first chapter of third unit is also done. Now coming on to the next chapter, which is human health and diseases. Give me a thumbs up, guys. Give me a thumbs up. All those of you who have completed these chapters with me, quickly let me know. Yes, 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 please. Clear? Let me know how many of you have completed all the chapters with me. Give me the number of chapters you have done with me. Yes, I want the number in the chat. I want the number in the chat. I want the number in the chat, guys. I want the number in the chat. 7. Only Sharanya has done all the chapters, I guess. I'm not going to stop till I complete class 12. I'm not going to stop till I complete class 12. Yes, please. 7. 7. Very good. Very good. All right, guys. Moving on to the next. Very good. Very good. All of you. Excellent. Excellent. Let's move on then. Let's see who stays till the end. Now that person will be the winner in life and in NEET. Yes or no? All right. Moving on to the next chapter, human health and diseases. See here. So, according to, yes. So, according to Ayurveda, health is a state of mind and body where there is balance of certain humors. humors. Okay? So this is what Ayurveda and Greek philosophers also believed in. Greek philosophers like Hippocrates and Indian Ayurveda believed in. So we believe that persons with black bile caused fevers and they have hot personality. Not like hot means they have hotness. They always feel a lot of heat in their body. Then this was discarded by, this theory was known as humor theory. This was discarded by William Harvey, where he explained there is nothing like that. So he disapproved. This theory was known as good humor theory, good humor hypothesis. But it was disapproved by William Harvey. Is that clear? He demonstrated the normal body temperature is not because of some humors or black bile. Then coming on to, yes, please. Okay, now health is affected by various factors, genetic factors, infections, lifestyle, right? And the term health is defined differently by different people, but simply does not mean the absence of disease or the physical fitness. It is a state of complete physical, mental, and social well being. This is how WHO defines health as. Is this point clear? Then, of course, balanced diet and personal hygiene, regular exercise maintain good health. And the diseases are of two types, guys. There are two types of diseases. The diseases are broadly infectious and non infectious. Infectious diseases are very common and they spread from one person to another, from one person to another. These are infectious diseases. While the non infectious diseases like cancer is a major cause of death. These NCERT lines are important. Then coming on to the next important part. So disease causing agent is known as pathogen. Mostly parasites, they are pathogens. Is that clear? Yes, please. So the first one, Salmonella typhi. It is a bacteria which causes typhoid. So it enters the small intestine through contaminated food and water. Sustained high fever up to 40 degrees Celsius, stomach pain, constipation, headache, loss of appetite, intestinal perforation, and even death might occur. So the classical case of typhoid carrier is typhoid Mary, or basically Mary Mellon. So Mary Mellon was a typhoid carrier. Now the typhoid can be diagnosed by this test, Vidal test. Very important test and very easy to remember. Other bacteria which are known to cause disease are Streptococcus pneumoniae and Haemophilus influenzae. Both are known to cause pneumonia. So basically what happens, see I've written the PYQs along. See, so basically what happens in pneumonia? So it infects the, which part of the body is infected in pneumonia? Alveoli. It gets filled with fluid, causing severe problems in respiration. So there are fevers, chills, cough, and headache. And lips and fingernails become gray to bluish. Are these points clear? Then other bacterial diseases are dysentery, plague, diphtheria. These are all bacterial in nature. Talking about the viral diseases, one very common viral disease is common cold, that is caused by rhinovirus. We also call it rhinitis. This is known as rhinitis by rhinovirus. Yes. It infects the nose and respiratory passage and not the lungs. These lines are important. What happens in common cold? Congestion, discharge, sore throat, hoarseness, cough, headache, and tiredness usually lasts 3 to 7 days and then it's fine. It spreads through droplets, so droplet infection, also by using the contaminated objects. Yes or no? Then coming on to the next disease, which is caused by a protozoan, that is malaria, caused by the protozoan known as Plasmodium. So we have 4 different species of Plasmodium: Plasmodium vivax, malariae, falciparum, and also ovale. NCERT has not written that, it is ovale. Okay, now different species cause different symptoms, different types of diseases. Are these points clear? Then, how does Plasmodium enter the human body? So, Plasmodium enters the human body in the form of a sporozoid. Remember, we have talked about sporozoans, special type of protozoans which have a special spore-like stage. This is that spore-like infectious form. Now, it enters through the bite of female Anopheles mosquito. enters within the liver cells, then attacks the RBC. So this is the order. First enters the liver cell, then enters the RBCs. The RBCs, when they rupture, they cause a toxic substance known as hemozoin, and then the malarial fever sets in. Are these points clear? Now, female mosquito Anopheles is a vector transmitting agent. Is this clear? So this is the cycle of mosquito. This is the cycle of mosquito. So mosquito bites and sends the sporozoites along with the bite. So parasites, that means the sporozoites, they reach the liver through the blood. So basically bites, it enters the blood, through blood it enters the liver, and then eventually from liver it is going to enter the RBCs. Is that clear? Now parasites will asexually multiply. How does it multiplies? Asexually multiply in the liver. The liver cells burst and released in the blood, and from blood it enters the RBC. Here again it reproduces asexually in RBC. Now the RBCs will burst and cause the cycles of fever. This is when the symptoms will appear. Are these points clear? Now, while the symptoms have already set in, the next stage is formed, which is known as gametocyte. This is the sexual stage. It is produced in RBC. So male and female gametocytes are produced, not the gametes. Please understand. So in humans, all this is occurring in the human. So in humans, it is not the gametocyte. Sorry, not the gamete. Remember, it is not gamete. It is the gametocyte. Now, when a fresh female mosquito will come for the blood meal, these gametocytes will be picked up. So female mosquito takes up the gametocytes with the blood meal. And now here gametes are formed inside the mosquito's alimentary canal or gut. Gamete formation occurs. Now these gametes are going to fertilize. After fertilization, they are going to form a zygote, and after zygote it is going to form a sporozoid, and these sporozoids are stored in salivary glands. So the mature infective stage, that is sporozoid, is stored in the salivary gland and escapes and migrates through the salivary glands. Are these points clear to all? Please let me know. Yes, guys. Then we have another another protozoan, which is Entamoeba histolytica. Right? This is a amoeboid protozoan, Entamoeba histolytica. The disease caused is known as amoebic dysentery 2021. So what are the diseases caused by protozoans in humans? Leishmaniasis, trypanosomiasis, amoebiasis. Yes or no? These are the three examples given in our book. So what are the symptoms of amoebiasis? Constipation, abdominal pain, cramps, stool with excess mucus, and blood clots. And what is the agent? Housefly. So it spreads through contaminated food and water. Again, contaminated food and water. So not just any contamination. See, contaminated food and water by fecal matter. When the contamination contains the fecal matter, that is the main source of infection for amoebic dysentery. Then another causative agent is Wuchereria. This Wuchereria is a roundworm, right? Then we have Ascaris, another roundworm. Both are Ascalminthes, and they are going to cause diseases. By the way, Ascarius cause ascariasis, and Wuchereria causes Wuchereriasis or filariasis. Let's talk about them one by one. See what happens during ascariasis. There is internal bleeding, muscular pain, fever, anemia, and blockage of intestinal glands. So these are the keywords. Blockage of intestinal passage is due to ascariasis. Again, contamination, food and water. Then Wuchereria. There are two species of Wuchereria, Wuchereria bancrofti, Wuchereria malai. Both cause elephantiasis or filariasis, right? There is blockage of lymphatic vessels of the lower limbs. It is a chronic inflammation. And see what happens. The leg becomes so swollen, just like an elephant's leg, hence the name. Then we have some fungal diseases. The fungi which are known for fungal diseases are Microsporum, Trichophyton, and Epidermophyton. And they are responsible for causing ringworms. So microsporum and trichophyton has already been asked multiple times. Maybe 2026 they are going to give value to epidermophyton now, right? So don't ignore this epidermophyton also just because I've not written any year along with it. So microsporum trichophyton has been asked multiple times. Maybe 2026 is about epidermophyton. So they cause diseases known as ringworms. Remember, most common infectious disease in one of the most common infectious disease in humans. Is it clear? And what happens in these diseases? Dry, scaly lesions. It affects skin, nails, and scalp. Is this clear? These are the common symptoms. Now, another common symptom is itching. Now, heat and moisture favors the growth of these fungi. Are these points clear? Are these points clear to all? Please let me know. Now, how do we actually control these? By using biocontrol agents, for example, cleaning, avoiding the stagnation of water, breeding places, right? The regular cleaning of household coolers, mosquito nets, and introducing the fish gambusia. So this gambusia is an example of biocontrol agent. Yes or no? We are using one organism against the other. Please let me know that these points are clear. Then also we can use, so what does gambusia actually do? It feeds on the mosquito larva. So you can also do that by spraying the insecticides in the ditches or drainage areas or swamps. Are these points clear to all? Yes or no? Then all these precautionary measures will not only control malaria but will also cause the control of diseases spread by Aedes mosquito. So basically Aedes mosquito spreads dengue and chikungunya. So they contain virus. So female Anopheles contains plasmodium, female Aedes contains these two viruses and cause these two diseases. Are these points clear to everyone? Now, there are certain deadly diseases like smallpox which have been completely eradicated by the use of vaccines, right? Other diseases are still trying to be, we are still trying to control them, like, see, polio has also been eradicated from India now, eradicated since 2012. Okay, but diphtheria, pneumonia, and tetanus, we are still working on them. Biotechnology is also getting us new drugs for the control. Coming on to the second very important aspect of immunity, that is, how does this immunity work? So first is innate immunity. Innate immunity is non-specific and since the time of birth, hence the name innate. It has four barriers. Physical barriers, which consist of mucus and skin, right? And mucus is present in respiratory, GIT, and urogenital tracts. Then the second barrier is physiological barrier. The physiological barrier consists of acid in stomach, saliva in mouth, tears in eyes. Clear? The third barrier is the cellular barrier. As the name says, it consists of cells called WBCs. WBCs like polymorphonuclear leukocytes. This term is used for neutrophils. Why? Because they can change their shape. Are these points clear to everyone? Please let me know. Are these points clear to everyone? Yes, guys. All clear? All right. Moving ahead then. All right. Moving ahead. Uh-uh. Now, there are other WBCs also which give us which provide us immunity. For example, your monocytes and macrophages and even natural killer cells. They all, so monocytes and macrophages, they are phagocytic. Natural killer cells are involved in killing your cancer cells, killing the cancer cells or tumorous cells. Not really cancer, tumor cells. Is this clear? Okay. Then we have fourth barrier as cytokine barrier. It produces special proteins. These are special proteins produced by virus infected cells. The cell which has already been attacked by the virus is going to produce this protein called interferon. And now it is going to protect the non-infected cells. So this is all innate immunity. So can you see that the number, oh sorry, the year in which all these questions have come up? Please let me know. All clear? Then the second type of immunity which we are blessed with is acquired immunity. So now, unlike innate, which was not pathogen specific, acquired immunity is pathogen specific. It is characterized by memory and only developed once we encounter the pathogen for the first time. So the immunity that develops when we encounter for the first time is known as the primary response. And the subsequent exposures will provide us with secondary and anamnestic response. So for the first time when we encounter the pathogen, we produce a primary response, while on subsequent encounters, the body produces a secondary or anamnestic response. And of course, secondary response is stronger and quicker. Is this clear? Why is it so? Because of the memory. Memory is a very important aspect. Now see, the two important cells which are involved in acquired immunity are B lymphocytes and T lymphocytes. Are these clear? The important cells are B and T lymphocytes. B lymphocytes they are going to produce special proteins in response to the pathogen, and these proteins are known as antibodies. Are these points clear to all? While each these antibodies, see, look at the structure of antibody. It has a Y-shaped structure. It has a Y-shaped structure, isn't it? Now there are the outer chains are smaller in size, so we call them the lighter chains. They are on the outer side, while the inner chains, they are known as the heavy chains. They are on the inner side. They are longer. So in lighter chain, do you see one variable, one constant? And then in heavy chains, we see one variable and three constant regions. All the chains are held together by disulfide bonds. Very important. C stands for the constant region and V stands for the variable region. Now, on the variable side, we have an antigen binding site. This also represents the N terminal of the polypeptide chain, while the constant regions have carboxylic group, so also known as C terminal. Are these points clear to everyone? Are these points clear to everyone? Now, talking about the second very important cell of immune system is the T lymphocyte. Now, immunity produced by B lymphocyte is AMIS because it produces antibodies, antibody mediated immune system, and and T cells provide us with cell mediated immune system. Is this clear? Now, some organs like heart, liver, kidney, it never fails to function satisfactorily. The transplantation is only remedy, right? But transplantation is not always accepted by the body. Why do we need a suitable donor? Because of this T lymphocyte. Okay, otherwise, if we don't have a suitable donor, then the grafts, that means the transplantation, will be rejected. It will be rejected. So the grafts would be rejected sooner or later if the matching is not done. Now this rejection is known as graft rejection. And the type of immune system responsible for graft rejection is cell-mediated immune system. And particularly, it is a T cytotoxic cell which is involved in graft rejection. Are these points clear? So it is written cell mediated is involved in graft rejection. It was in 2022. This line was in 2022, 19 and 17 in NEET exam. Are these points clear to everyone? Then guys, let's talk about the two types of immunity. Again, very important chapter. See here. So active and passive immunity. When a host is exposed to antigen, antibodies are produced. So if the body is able to produce antibodies on its own, this is known as active immunity. It is slow but gives off full effective response. Is that clear? But in times of emergency, when ready-made antibodies are provided directly, are directly given, this is known as passive immunity. Is this clear? Now, example of active immunity is clinical exposure. When you just acquire the disease on getting exposed to someone, clinical exposure. Also, when you inject vaccines, vaccines also provide active immunity. While in case of passive immunity, we have one example of colostrum, mother's milk. So we know mother's milk has an antibody called IgA. So you're giving a ready-made antibody to the child. So since the baby is getting a ready-made antibody, so immunity provided by colostrum is an example of passive immunity. Is this clear? This is an example of passive immunity. Similarly, fetus can also get some antibodies directly from mother through placenta. This is also another example of passive immunity. Similarly, we have some formulations like anti-venom, that is also passive immunity. Is this clear? These are all examples of passive immunity. Please let me know that these points are clear. Now, talking about the vaccination and immunization. See, first of all, you must remember that principle of immunization is based on the property of memory. This memory is very, very important. Is this clear? Now, in vaccination, what do we give? We actually give the pathogen, antigen, protein of the pathogen, or the inactivated or weakened of pathogen. Is that clear? Antibodies produced in the body against the antigen would neutralize the pathogenic agent. They generate memory B and T cells. Is that clear? B memory and T memory cells. So, see, it is written, even in cases of snake bites, the injection which is given to the patient contains pre-formed antibodies. So now see, when we are giving an inactivated pathogen or the protein of the pathogen, this is known as active vaccination or active immunization. This is known as active immunization. But when we are directly injecting the ready-made antibodies, that is known as passive immunization. Please let me know that these points are clear. So this passive immunization helps in quick immune response. Is this clear? If the person is already infected, if the person is infected with some deadly disease, in order to give quick immune response, you always give passive immunization. For example, in case of tetanus or in case of snake bite. Are these points clear? Are these points clear to all? Please let me know. Now, rDNA technology is also helping us a lot. So, you've already read this chapter in the production of antigenic polypeptides of pathogen in bacteria and in yeast. So, vaccines which are produced by the help of rDNA, these vaccines are called the third generation vaccines. So, rDNA vaccines, one such example is vaccine against hepatitis B. So rDNA vaccine is hepatitis B vaccine, and the host used during the formation of this vaccine was or is a yeast cell. Please let me know that all these points are clear. Please let me know all these points are clear to all. Now guys, talking about allergy. Talking about allergies. See, what is an allergy? It is an exaggerated response of the immune system against a certain antigen. Is it clear? Now, certain antigen to which this immune response is generated, these certain antigens are known as allergens. So, antibodies which are produced during allergy are called IgG. What are the common allergens? Dust, pollen, animal dander. This was a question in 2024. Then, which are the other chemicals which are produced during allergy? It is histamine and serotonin from the mast cells. Is this clear? So for treatment, what do we give? The treatment of allergy involves the use of antihistamine, adrenaline and steroids. They reduce the symptoms. Is this clear? Treatment. Yes or no? Please let me know all these points are clear to all. So one such example of these allergies are asthma, right? Okay. Moving on to another type of disease, which is autoimmunity. Higher vertebrates based on their ability to differentiate between the foreign organisms from self-organisms. Now, when we fail to differentiate this the failure to do this leads to autoimmunity. Are these points clear? So what happens in autoimmunity? Body attacks the cells. So examples, rheumatoid arthritis. There are many other examples which I've written here because they were asked in different years. Systemic lupus or erythematosus. This was in 2024. Myasthenia gravis, psoriasis, vitiligo. Although psoriasis and vitiligo were asked long time back, but rest of the examples are in recent years. Then we have lymphoid organs which are going to produce lymphocytes for us. Yes or no? So after maturation, see, after maturation, the lymphocytes migrate to secondary lymphoid organs. So the primary ones are only two, bone marrow and thymus. Bone marrow and thymus, while the secondary ones are spleen, lymph nodes, tonsils, Peyer's patches of small intestine and appendix. So uh, what is the role of secondary lymphocytes? Basically, they provide the site for interaction of lymphocyte with the antigen to become the effector cell. Are these points clear? The bone marrow is the main lymphoid organ. It is the primary lymphoid organ where blood cells, including lymphocytes, are produced. Now, both bone marrow and thymus, these bone marrow and thymus, these are the primary structures. They provide microenvironments for the development and maturation of T lymphocytes. Is this clear? Also, guys, spleen is a large bean-shaped structure and also the largest lymphoid organ, contains these phagocytes and lymphocytes, filter of the blood, and large reservoir of RBCs also, and also the graveyard. We have read about it yesterday. It is not just a reservoir, it is a graveyard also. So then we have lymphoid tissue, which forms the major part. So this is the maximum secondary lymphoid organ present in our body. That is the malt. Malt. More than 50%. This is the malt. Mucosa associated lymphoid tissue present in respiratory, digestive, and urogenital tracts. And as written clearly, it constitutes more than 50% of the lymphoid tissue in the human body. Are these points clear? Now coming on to the next disease, which is AIDS, acquired immunodeficiency syndrome. The virus is HIV, which is a retrovirus because it has RNA genome enclosed within the envelope. Is that clear? Now we know how the disease spreads. So basically now RNA, there are two identical molecules of single-stranded RNA in HIV, and also there are two molecules of reverse transcriptase. Coming directly here to the cycle. Yes, please. Look here. So RNA containing virus. So this HIV or the retrovirus has RNA. Is that clear? Now virus is going to infect the normal cell. Usually it is a macrophage because macrophage goes to ingest it. So once macrophage ingests it, the virus enters the cell. This viral RNA is now going to change into the viral DNA. RNA becomes a viral DNA using the process reverse transcription and enzyme transcriptase, reverse transcriptase. Is this clear? Then what happens? Once the DNA is formed, this viral DNA is going to be incorporated into the human genome, human DNA. Right? This is the human DNA, and let's say this is where the viral DNA also adds up. Now this new viral RNA, this new recombinant DNA is produced. This rDNA is going to produce new viral particles and new viruses are produced like this. So therefore, the animal cell acts as a factory cell for HIV. Is that point clear? This is how things happen. Please let me know that these points are clear. Please let me these points are clear. Now see, now ultimately macrophage continues to produce virus and this way HIV factory written. HIV enters the T helper cells, replicates and produces progeny viruses. So second cell which is affected by the HIV is a T helper cell. Is it clear? Now the diagnostic test we have already discussed is a ELISA, enzyme linked immunosorbent assay. But in case ELISA is positive, it is confirmed using Western blotting. Confirmed by Western blotting. So if ELISA positive, then we go for Western blotting. Are these points clear? Please let me know. Please let me know these points are clear to all. Now coming to AIDS, so there is the special body, National AIDS Control Organization. It's an NGO doing a lot to educate people about AIDS. Is that clear? Coming on to cancer, see, there are some properties of the cancer cells. So unlike normal cells which show contact inhibition, normal cells will show contact inhibition, nor cancer cells are very loosely placed. Is it clear? So cancer cells, they don't have this property. Also, they show uncontrolled growth. Clear? So contact inhibition basically inhibits the uncontrolled growth, but when normal cancer cells lose this property, so they divide in an uncontrolled manner. Clear? And they form masses of cells called tumors, which can be benign or malignant. So benign are confined to the original place, while malignant they spread. So they invade and they damage the surrounding tissue. This invasion, this property of invasion is known as metastasis and the most feared property. So once the cancer becomes metastatic, that it's very difficult to control and treat. Are these points clear? Please let me know. Please let me know all these points are clear to you. Next, guys, we have the causes of cancers. So we have various causes. For example, all the causes ultimately are known as carcinogens, right? They could be physical, chemical, or biological in nature. So what are the physical agents like? Physical agents means radiations like ionizing radiations are X-rays, gamma rays. Okay. X-rays and gamma rays are the ionizing ones, and non-ionizing are the UV radiations. So you should be very clear with this. Ionizing are the X-rays and the gamma rays, XY, and then non-ionizing are the UV ones. Is this clear? New. Next, we have chemical carcinogens also. For example, in tobacco smoke is a major cause of lung cancer. Then we have biological agents also. So biological agents consist of the oncogenic viruses. And then they have these special genes called oncogenes. Normally, these genes are inactive in our body and we call them proto-oncogenes. So this is the inactive form. So till it is inactive, it is all good for us. But once they become active, it is dreadful and leads to cancer. Then cancer detection, guys, is due to how do we detect cancer? So we have multiple tests, for example, biopsy, we have histopathological studies, we have blood and bone marrow test, and for the increased cell count in case of leukemia. Is it clear? So bone marrow test is done for leukemia. Are these points clear till now? Then we have some other histopathological studies or techniques like radiography. In radiography, we use X-rays and then we have CT scan, we have MRI scan. All these methods help in detecting cancer. So in CT scan also we use, that is computed tomography, we use X-rays to get the 3D image. So that is why CT scan is more precise as compared to a normal X-ray. While in MRI we use magnetic field and non-ionizing radiation, very strong magnetic fields are used. Then we have certain antibodies which are cancer specific to detect different cancers. Then there are certain molecular techniques of molecular biology also, like autoradiography. Is that clear? Which detect the genes, whether they are in the active form or in the inactive form. Please let me know that these points are clear. Then, guys, talking about the treatment. Let's talk about the treatment. So the treatment occurs at 4 levels. So we have surgery, radiotherapy, immunotherapy, and the chemotherapy. So chemotherapy involves some drugs which kill the cancer, right? Now side effects of these drugs are hair loss and anemia. Now combination of all these methods are used to treat cancer. Now talking about the talking about the immunotherapy. This is immunotherapy. So in immunotherapy, basically we use alpha interferon to destroy the tumor. Is this point clear? Coming on to the last part, drugs. So opioids. See, opioids produce drugs from opioids are the drugs from opium. That the opioid receptors are present on CNS and GIT. Are these points clear? Very important. Where are the receptors present? GIT and CNS. One example of opioid is heroin, also known as smack, chemically known as diacetyl morphine. Is that clear? It is produced by diacetylation of morphine and extracted. Where is the main chemical, that is opium, obtained from? Opium is obtained from the latex, extracted from the latex of this poppy plant. The scientific name is Papaver somniferum. And heroin, like any other opioid, is a depressant which slows down the body actions. Is it clear? It is a depressant. Then we have cannabinoids, which interact the cannabinoid receptors present principally in the brain. Natural cannabinoids obtained from the inflorescence of the plant, that is cannabis sativa. Now, flower tops, leaves, and the raisins of cannabis plants are used in the various combinations to produce marijuana, hashish, hash, charas, and ganja. So, second, is that clear? So, all these cannabinoids, they have effects on cardiovascular system, very bad effect on your heart health. Now, see the shape of the leaf of cannabis. Is it clear? And now the third drug that we are going to talk about is cocaine. Now, cocaine is obtained from a plant, Erythroxylum coca, which is a native of basically South America. And it interferes with the neurotransmitter dopamine. Is that clear? Now, it is commonly known as coke or crack and produces a sense of euphoria. It has a stimulating action. So, basically, it is a stimulant. It is a stimulant. Is this clear? Excessive dose can cause hallucinations. See, opioid is a depressant, cannabinoid is a hallucinogen, while cocaine is a stimulant. Is this clear? But excessive use of cocaine can be hallucinogenic. Now, other sources of the other plants which can have hallucinogenic properties are Atropa belladonna, and the second is datura. So, this picture is a datura. Is this clear? These days, cannabinoids are being abused by many sportsmen. Now, there are other synthetic artificial drugs also, barbiturates, amphetamines, and benzodiazepines, which are actually used as medicines but have been misused also and the medicinal uses to cope up with diseases like depression and insomnia. Now, morphine is used as an effective sedative and a painkiller used for patients who undergo surgery. So, it is a painkiller. Is that clear to all? Is that clear to everyone? Please let me know. So, guys, this was the most important part of this chapter. And with this, we move on to the next chapter, which is chapter number 6. Please let me know how many of you have completed this with me. All of you who have completed this with me, how many chapters? Give me a thumbs up. Yes, please. We are not going to stop till we complete complete the syllabus. Quick, quick, quick. Give me a number in the chat. How many chapters have you completed with me? See, I'm already going slow now because of my throat. Yes, please. Quickly let me know. How many chapters have you have you read with me now? Till now. How many chapters? Yes, please. We are going to start with another unit, the second unit of this book actually. But for us, it is the third unit of the night. Yes, guys. How many chapters have you finished with me? 7? 7? It's still 7? It's only Yuvraj who's writing. Yes, how many chapters, guys, have you finished with me? Let me know in the chat. So we are going to start with the chapter of evolution. All very good, Ansh. Very good. All right, coming on to this chapter. So, see, please note that NCERT has made this change. The origin of universe is in 13.8, was almost 13.8 billion years old. And universe, the origin of universe is explained by Big Bang theory, right? So it was hydrogen and helium formed sometime later and led to the formation of galaxies and then Milky Way and Earth. So the formation of galaxies or Earth happened 4.5 billion years back. And then talking about life, Life appeared 500 million years after the formation, that is 4 billion years back. Please let me know all these points are clear. Now, there is a certain sequence of events given in the book in a haphazard manner. So, I have compiled it up. See here, sequence of events during the origin of life. This was question 2016. So originally, there were organic monomers, then organic polymers, protobions, and DNA-based genetic system. So there were other theories also. Greek thinkers believed in spores coming from different planets, and these spores were known as panspermia, right? Then there was this theory known as spontaneous generation, which believed the life came from decaying and rotting matter. Then later on, it was Louis Pasteur which explained that life came from pre-existing life. He used this experiment, round bottom flask experiment. Is that clear? But the theory which has maximum support is given by Oparin and Haldane. Why? Because there is some experimental evidence for that. So these people believed that the first form of life came from pre-existing non-living matter like RNA and protein. And lit, so there has been a chemical evolution. That is why this theory is known as chemosynthetic theory. So it believes in chemical evolution of life. Now the conditions of Earth during this was high temperature, volcanic storms, reducing atmosphere. And in 1953, we found a S.L. Miller, Urey and Miller basically, Urey and Miller, they gave us an experimental evidence. So in this, there was an electric discharged in a closed flask, and in the flask in 2020, see the question has been asked. What are the four molecules used during the experiment? It was methane, ammonia, then water and hydrogen. So water was taken at 800 degrees Celsius, and in this experiment, formation of sugars, nitrogenous bases, pigments, and fats was observed. Are these points clear? Are these points clear to all? Please let me know. See, this is the experimental setup. So spark discharge, these are the four molecules taken, and discharge of 15,000 volt was created, and after 17 or 18 days, we acquired, see, what did we find? We found that there was sugar and nitrogenous base and pigments and fats formed. Please let me know all these points are clear to all. Now, Earth is about 4,000 years old. So, this was a conventional belief, not the real thing. This was a conventional belief. All right. Yes, please. Wait a second. Wait a second. Yes. Let's come directly to the previous year questions here. Yes, yes. Okay. So, the most important aspect from this chapter is the examples of homologous and analogous organs. Guys, see here. So, whale, bat, cheetah, and human. So, they all share similarity in the pattern of bone of four limbs. So, this is an example of homologous organs. So, for homologous, the trick to remember is they are similar in origin, similar structure, but different function. So, whenever there is a set of organs given to you, so just see if they are performing different functions, they are homologous to each other. Okay, so different functions, although they have similar anatomical structure, the same bones. And these homologous organs, they show divergent evolution. So we remember HDAC. Homologous organs show divergent evolution, and analogous organs are going to show convergent evolution. So the trick is HDAC. Is it clear? So homology indicates the common ancestry. Is that clear? Then wings of birds and flippers of whales. Again, example of homology, modified forelimbs. Bougainvillea and cucurbita, thorns and tendrils. Again, homology. Is this clear? Then coming on to analogy. Analogy is exactly opposite. So in analogy, the functions will be same. Same functions indicate analogy. So wings of butterfly and birds. Is that clear? And analogous organs always show convergent evolution. See, flippers of penguin and dolphin. Now, penguin and dolphin, they show analogy. Then sweet potato and potato. One is the root modification, one is the stem modification, forming the same function. Are these points clear to all? Are these points clear to everyone? Please let me know. So, then we have this another example. Excess of herbicides and pesticides have led to the resistant varieties. This is an example of this is an example of evolution, modern evolution through anthropogenic action. Anthropogenic means because of human action. Is this clear? See here. So drug resistance, man-made dog breeds, these are all anthropogenic actions and examples of modern evolution. Now, the variety in the beak of finches, Darwin finches, found in the Galapagos Island, this represents adaptive radiation. Another example of adaptive radiation is in case of marsupials. Is this clear? Is in case of marsupials. Yes, guys. This is in case of marsupials. So you have to learn all these names. So examples came in 2023. Please see here. All the names are important. You must know. The question can be very simple like this. Which of the following does not belong to the marsupial radiation? So you have to remember all the 10 names. Is this clear? And then you have to remember this is an example of convergent evolution or adaptive convergence. This is an example of adaptive convergence. Now, adaptive convergence is exhibited by placental mammals and Australian marsupials. You have to remember them in pairs. Mole and marsupial mole, anteater and numbat. See, this came in 2024. Then lemur and spotted cuscus, flying squirrel and flying phalanger, and then bobcat and Tasmanian cat. Is that clear? So this was the example. Then coming on to the mechanism of evolution. So how evolution occurs was first told by or only mentioned by Hugo de Vries. So Hugo de Vries said the mutation is the cause of evolution. So mutations are always random and directionless. But according to Darwin, the evolution is small and directional. So compare what are the differences. So De Vries believed that mutation causes speciation, that is formation of new species, and it requires a single step large mutation which is known as saltation. Is this clear? Then moving on to Hardy-Weinberg equilibrium. Now this is a numerical based topic. You get numericals also. So what is Hardy-Weinberg equilibrium? It says that allelic frequency, allelic frequency in a population is always stable or constant from generation to generation. That means the gene pool will always remain constant, but under 3 conditions only. Now the conditions are that the population, see what are the conditions for the Hardy-Weinberg equilibrium to apply. Number 1, it has to be a large population. It has to be a large population conditions for Hardy-Weinberg equilibrium. Large population, random mating, right? Large population, random mating, and then there has to be no migration. Now see here. So the formula for this is P plus Q is equal to 1, where P is the allelic frequency of dominant allele, Q is the frequency of recessive allele. Is it clear? Now on squaring we get P square plus 2PQ plus is equal to 1. Now this is the binomial expansion, right? Now P square is the frequency of homozygous dominant organisms. 2PQ is the frequency of heterozygous organisms, and Q square is the frequency of homozygous recessive organisms. Are these points clear? Now, there are 5 factors which are going to disturb the Hardy-Weinberg equilibrium. Gene migration or gene flow, genetic drift, mutation, genetic recombination, and natural selection. These are the 5 factors. Very, very important. Now, new genes or alleles, if added to the new population, then they are lost from the old generation. This is called the gene flow or the genetic drift. That means change in the allelic frequency would take place. Now, the drifted population becomes the founder of the new species, and this is called the founder effect. This is called the founder effect. Now, directional changes, guys. What are the directional changes here? So, more individuals acquire value other than the mean value. This is called the directional change. Now, see here, we have brief accounts of evolution. We have brief accounts, very, very important. Earliest organisms were non-green anaerobes. First autotrophs were chemoautotrophs who never released oxygen. Let's come back to this. Evolution of plants. Now, see here. You have to understand that which plant was evolved during which phase. For example, Zosterophyllum in Silurian, lycopods came in Permian, then ferns, conifers, and cycads came in Jurassic, and so did the dinosaurs. So all came in Jurassic. Then you have to see that ginkgo, nettles, and dicots, they have the origin in Cretaceous. So you have to learn them like this: Cretaceous. And angiosperms and bryophytes, they have origin in Cenozoic era. Please let me know that all these points are clear. Then very important line which came in 2024, that South Africa happens to have a coelacanth, which is a lobe fin, and it is a there's a connecting link. See, fish to amphibia. So we say that lobe fins were evolved into first amphibians, and they lived on both land and water. So who's the ancestor of amphibian? Lobefin. One example of lobefin is coelacanth. Is this clear to all? Then giant pteridophytes. They were present, but they all fell to form the coal deposits. So coal deposition, coal is majorly produced by these pteridophytes. Now see, so reptiles went back into the water and evolved fish-like reptiles 200 million years ago. That is why Ichthyorus is a fish-like reptile. Tyrannosaurus, 20 feet, dagger-like feet. About 65 million years ago, dinosaurs disappeared due to some climatic changes. Now, the first mammals were like shrews. Is that clear? Mammals were viviparous, protected, unborn, were protected inside the mother's body. Is this point clear to all? Please let me know. And then, due to some continental drift, the pouched mammals of Australia, they were separated, and due to lack of competition, they just survived as such. Please let me know all these points are clear. Coming on to the evolution of man. 15 million years ago, primates called Dryopithecus and Ramapithecus existed. So first was the common ancestor of humans and apes, that is Dryopithecus. Then came Ramapithecus. Both were hairy and walked like gorillas and chimpanzees. Ramapithecus was more man-like, Dryopithecus was more ape-like. So origin in Africa. See, we discovered the fossils in Ethiopia and Tanzania. So origin of man or human in Africa. Is this clear? So then we believe Australopithecus came up in East Africa. So Australopithecus walked upright. First one to walk upright was Australopithecus. Is this clear? Then came first human-like hominid was Homo habilis. See the brain capacity of Homo habilis, 650 to 800 cc. Then this Homo habilis gradually moved to Java, and we call this phase as Homo erectus. 1.5 million years ago, and the brain size increased to 900 cc. So see, I have written all the stages of human evolution here. They were asked in 2024 along with the cranial capacities. Then after Homo erectus came Neanderthal man with the brain size 1400 cc. And these used to cover their body with hides, animal skin, and buried their dead body. Is it clear? And eventually came Homo sapiens. During the Ice Age, between 75,000 to 10,000 years ago, Homo sapiens arose. Is this point clear? Is this point clear to all? So guys, with this, we complete the chapter of evolution. I hope you are revising all the PYQs with me. Yes, please. Are we revising all the PYQs with me? How many of you have completed this chapter with me? How many of you have completed this chapter with me? Yes, please. No, we can't afford a break. We have many more chapters to complete. See, I'm already going slow now. Yes, please. All right. Moving on to the next chapter. What is this? Sorry, this is by mistake. Moving on to the next chapter, genetics and evolution. Principles of inheritance. Let's come directly to the PYQs here. Okay. So first of all, you must know which were the characters studied by Mendel. The characters studied by Mendel. See, 2022 and 2017. So sometimes even easy equation is also asked. So for 7 years, Mendel studied. What is the year in which he studied? 1856 to 63 and proposed the laws of inheritance. This was a statement based question. Then the 7 contrasting features studied by Mendel. So you have to remember what were the contrasting features. Clear? Then Mendel chose how many true breeding pea plants? 14 true breeding varieties of pea plant. Is that clear? How many? 14. Then moving on to, wait, just a second. Yes, guys, just a second. It is taking a while for the file to be loaded. Let me open with the other file till then. Wait. Wait a second. Why have I downloaded this fifth file? Okay, wait a second. Yes, give me a second, guys. Wait a second. By the time this opens, let me continue with the chapter. Okay, so where are we? So we are done with this. So then there is another line that came up in 2024. Genes which code for a pair of contrasting characters are known as alleles. So the genes coding for contrasting traits are known as alleles, like capital T and small t. It is taking quite a while to be loaded. I don't know why. Quick. I think we can start. Wait, we can maybe take another chapter also. Fifth is not there. Sixth, we are done actually. Now we need chapter number 4. Sixth is done. Fifth, and then we have chapter number 3. Where is it? Yeah, this is chapter number 3. And finally, we have chapter number 1 here. Yeah, let's see. Okay. Next is the production of gametes by the plants, by the parents. The formation of zygotes F1 and F2 plants can be understood through a diagram known as the Punnett square. So the diagram which helps us to understand the production of gametes by the parents easily. This was given by a British geneticist, Reginald C. Punnett, and that is why we call it Punnett square also, right? Then coming on to the genotypic ratio of monohybrid cross. Monohybrid cross was 1 is to 2 is to 1. This is the genotypic ratio, while this is the phenotypic ratio. We know that already. This is easy. So sometimes easy questions are also asked, guys. See, now coming back to 2024, we have test cross. So in test cross, a dominant phenotype can be crossed with a recessive parent to check whether the dominant phenotype is because of homozygous condition or heterozygous condition. Is that clear? Now, in order to determine the genotype of the dominant trait, we have test cross. Is that clear? So test cross is going to clarify whether the tallness is because of capital T or small t, or similarly for every other phenotype. Now, what is a back cross? Back cross is a cross of F1 generation with any parent. So see, if we are crossing it with homozygous recessive parent, it becomes a test cross. But if it is any parent, it is a back cross. Is it clear? So test cross is a back cross. Test cross is a back cross, but every back cross is not a test cross. We are clear? Okay. Now, similarly, so it is given here that black dominant over the white. Find the genotype of black. So how do we find the genotype of the dominant trait? So again, it is through test cross. Is this clear? So this is the significance of test cross. So what is this? What does law of dominance say? This is the question of 2024. So characters controlled by discrete units called factors. Factors occurs in pairs, and one pair dominates over the other, which is known as recessive. Is this clear? So expression of only one parental character is seen in the monohybrid cross of F1 generation. Are these points clear? Then guys, 2024 also asked us about incomplete dominance. So instead of red and white, we got pink and red. So what will be the phenotype of progeny if one pink? That means RR is uh, crossed with RR. So only in the homozygous condition we get R, and in the heterozygous condition we find pink. So this is incomplete dominance when a dominant trait is only produced during the homozygous condition. Now the case study of incomplete dominance is a snapdragon or anti-rhinium. Is this clear? Now in a cross, see here, in a cross, let's quickly see, just a second. Hanji, in a cross between true breeding red colored flowers and the white colored flowers, we get F1RR. Now this is not red, it is pink. So in case of incomplete dominance, you have to see that phenotypic ratio gets modified and it is just equal to the genotypic ratio. So instead of 3:1, we get 1:2:1, and so is the genotypic ratio as well. This is your antirrhinium or snapdragon showing incomplete dominance. Now basically the modified allele here is responsible for the production of one is normal or less efficient enzyme, non-functional enzyme, or no enzyme at all. Are these points clear? Then talking about codominance, the example is ABO blood group system. Then ABO inheritance shows three different concepts. One is multiple allelism because there is presence of three different alleles. IA, IB, and small I. It also shows us dominance. So A and B blood group is because of dominance, while complete dominance, right? While AB blood group is because of codominance. So there are three phenomena which are exhibited during ABO blood grouping. Are these clear? So it is a numerical based question also, where you can find out if the father has this blood group, mother has this, what is the blood group of the baby, and so on. Now similarly, see how many genotypes and phenotypes are possible in such case. So otherwise, it's very clear that there are three different alleles and six different combinations from these different alleles are possible, and six different genotypes. Total six different genotypes of the human blood groups tables are possible. And how many phenotypes? Only four. Phenotypes are the number of blood groups. So genotypes would be 6 and phenotypes would be 4. Is that clear? Now you can cross any genotype and find out what will be the phenotype. Now, in case of starch synthesis, now this is very, very important. So there is a single gene which has more than one effect. Now this is known as pleiotropy. For example, see here. Yes, so this is the B gene. Now this B gene is responsible for the shape of the seed as well as the grain size, starch grain size. Starch grain size. Now see, when there is this condition, homozygous dominant condition, so shape will be round, but starch grain will be large in size. Even this, in heterozygous condition, shape would be round, but starch grain would be intermediate, right? And then in homozygous recessive condition, only here the shape will be wrinkled and starch grain would be very small in size. Are these points clear? So this is what is given in this paragraph. Now coming on to inheritance of two genes. So in inheritance of two genes, Mendel gave us another law, that is law of independent assortment, and the ratio of phenotype is 9:3:3:1. Are these points clear to everyone? Please let me know that these points are super clear. Then moving on to this very important paragraph of chromosomal inheritance. Wait. Yes. So who gave us this chromosomal theory? Chromosomal theory of inheritance was given by Sutton and Boveri, and it was the experimental verification of chromosomal theory was given by, yes, who gave the experimental verification? Quickly let me know. It was Thomas Hunt Morgan. Thomas Hunt Morgan. So Morgan gave a experimental verification and used the organism Drosophila melanogaster. So why was Drosophila melanogaster or the fruit fly only used? So the reasons were that they can be grown on simple synthetic medium. Number one, life cycle is only of 2 weeks. So easily and shorter life cycle. Then single mating gives us a large progeny. This is the third reason. Fourth, there is clear distinction between male and female. So sexual dimorphism is there. And then hereditary variations can be seen under low power microscopes. So that is why we have chosen this Drosophila. Then coming on to two genes that did not segregate during division. So this is because of another phenomena known as linkage. This was discovered by Morgan. So otherwise they are supposed to segregate, but if they don't segregate independently, this is against the Mendelian law, and this is known as linkage. So when two genes are situated on the same chromosome, that is when they show linkage, and this term and phenomena was discovered by Morgan. Are these points clear? Now, on the basis of recombination frequency, the student, he was a student of, I think, Morgan, yes, Alfred Sturtevant, he said that frequency of recombination between the gene pair is equivalent to the distance between the genes. So if you are given a distance between A and B equal to 3.4 centimorgan, so in that case, you know the frequency, the recombination frequency will also be 34%, or the vice versa. So if you have recombination frequency, you know the distance. If you have the distance, you can easily find the recombination frequency. And with the help of this, he was able to map the position of genes on the chromosome and produce genetic maps. Now the unit used for mapping is centimorgan. So basically he gave a tribute to his teacher, that is Morgan. 1 centimorgan is equivalent to 1% crossover. Are these points clear? So see, in these two crosses, these are very important. In these two crosses, cross A and cross B. So in one case, we have taken the body with the see here, in this cross, what do we wait in this cross, what do we see? In this cross, we see the parental type shows 98.7% frequency and recombinant is only 1.3. So this is a case of linkage. While when the parental frequency is 62 and recombinant is 37.2, this is a case of independent assortment. So actually, assortment. Is this clear? So based on the genetic frequencies, you can easily find out whether the two genes are showing linkage or independent assortment. Then we have this case of polygenic inheritance. Now this is again a non-Mendelian pattern. So the example of polygenic inheritance is seen in case of human skin color, which is controlled by 3 different genes, A, B, C. And as a result, there are 3 genes and 6 alleles. So in this case, it is not which gene is dominant, we just count the number of dominant genes. So the genotype with 3 dominant genes and 3 recessive genes will be of intermediate. So if we have 6 dominant, maximum melanin will be produced, it will be maximum dark or black color. If there is no dominant gene, no melanin will be produced. So we are not going to see which is the dominant gene, we are only going to count the number of the dominant genes. Are these points clear to everyone? Please let me know. Then we have just studied this phenomena of pleiotropy above, where we discussed the B gene showing multiple effects. It is B gene was affecting the shape of the seed, as well as B gene was affecting what? The starch grain size. So one gene and multiple phenotypic expressions is pleiotropy. Now there are other examples of pleiotropy, phenylketonuria. We have phenylalanine. So in phenylketonuria, this enzyme is not produced, right? Other example of pleiotropy is sickle cell anemia also. Sickle cell anemia. We'll just talk about it. Then phenylketonuria is characterized by mental retardation, skin pigmentation, and reduction in hair. Then very important topic is sex determination. In insects, for example, grasshopper, the male is exotype and female is XX type. Is this clear? Then in grasshopper, see, we have already discussed this. It came in 2022. Just a second, guys. Just a second. All right. Okay. Why is it not getting loaded? Let me check with the other chapters. No, they are fine. They are just fine. This chapter is a bit heavy, I guess. Yeah. Okay. So we see here, now see, coming here. Yes, grasshopper has XX exotype, right? So remember the underlining statement that it is always the heterogametic partner which is going to determine the gender of the baby. Now similarly in case of Drosophila as well as in humans, the heterogametic male. So humans and Drosophila has XXXY type, where XY is the male, right? So male is going to determine the gender. Now these are examples of male heterogamity. Even in grasshopper and in Drosophila and in humans, it shows male heterogamity. But in case of fowls or birds, there is female heterogamity. There is female heterogamity. Do we see that? So here, what happens? So the female, see, the female is going to be female is going to be ZW, while male is going to be ZZ. Is this clear? So whoever is heterogametic is going to determine the sex of the baby. So this is already discussed. While another method of sex determination as seen in honeybee is known as haplodiplontic method. We call it haplodiplontic method because here the number of set of chromosomes is going to determine. For example, male has single set of chromosomes and female has diploid set of chromosomes. Is this clear? So female is going to produce eggs by meiosis, while male is going to produce gametes or sperms by the help of mitosis. Is this clear? Now how does this so how is it decided? So basically, if the female egg gets fertilized with the sperm, it is going to result in 2n, which is again female. But if the egg is not fertilized, so non-fertilized egg will result in the formation of male. So basically, non-fertilized egg when gives rise, it is known as parthenogenesis. So males are always produced by parthenogenesis. Are these points clear to all? Are these points clear to all? So guys, next, this table is very important. You have to learn the symbols. So this symbol was asked in 2023, consanguineous mating. That means mating between the relatives. So there are cultures where brother sister get married. So this is consanguineous mating. Is that clear? So you must know the symbols here. So what is pedigree analysis? Pedigree analysis is going to give us analysis of traits in several generations of a family. So basically it is a family tree over generations. Is it clear? And it helps us to trace the inheritance of a specific trait or an abnormality. Is that clear to all? So it is a numerical based topic. I'll be taking a separate class in unit wise revision where we will be discussing these numericals. Are these points clear? So this was a pedigree based question asked in 2025. Now you must know that which diseases are autosomal dominant, which is the example is myotonic dystrophy, while autosomal recessive diseases are sickle cell anemia and hemophilia. Is it clear? Now, both are blood disorders. Similarly, we have autosomal dominant diseases also and sex-linked dominant diseases as well. So, how do we detect it? We will see it in the unit-wise revision. Are these points clear to all? So, see. So, hemophilia is one of the X-linked recessive disease. So, what basically happens here? So, in hemophilia, the blood stops clotting properly. It doesn't clot properly. So, excessive bleeding, non-stop bleeding will take place. Now, heterozygous female is only a carrier. In females, it only occurs in the homozygous condition. Only female with both XH or recessive genes will have hemophilia, while the heterozygous female, see, while the heterozygous female, that is XHX, she's only a carrier. Is that clear? But in males, even since males are heterozygous, they have X, only single X uh, chromosome. So the hemizygous condition will lead to the disease. Are these points clear? So it is known as a royal disease also because Queen Victoria's whole family has had this disease. Then talking about the sickle cell anemia, it is an autosomal linked recessive disorder. So what really happens here? What really happens in this faulty hemoglobin is formed. So the beta chain of hemoglobin, the sixth amino acid in the beta chain, is substituted. So in the normal hemoglobin, it is supposed to be glutamic acid, but instead of glutamic acid, we have valine in place of that. So sixth one is substituted with valine. Now see, the mRNA that codes for glutamic acid is GAG, but when it becomes GUG, it starts coding for valine. Are these points clear to all? Are these points clear to all? Is that clear? Then coming on to the next disease is phenylketonuria. Already discussed the symptoms. It is an autosomal recessive disease. Similarly, thalassemia is an autosomal recessive disease. Now, there are two types of thalassemia, alpha and beta. So, alpha is when alpha chains of hemoglobin are not produced, and beta thalassemia when beta chains of hemoglobin are not produced. Are these clear? So, alpha thalassemia occurs when because alpha chain is produced by these two genes, so it occurs due to abnormality in these two genes with HbA1 and HbA2. These genes are found on chromosome number 16. While beta thalassemia, the gene for beta thalassemia, which is HBB, is present on chromosome number 11. Is that clear? Now, this is an important line. The thalassemia is a quantitative disorder, while sickle cell anemia is a qualitative disorder. Because in thalassemia, the globin chains are not formed, either alpha or beta or both. But in sickle cell anemia, both are formed, but the faulty version of it. Are these points clear? Then coming on to chromosomal disorders like aneuploidy, where there is a of extra copy of gain or loss actually. Aneuploidy is gain or loss of a chromosome. Is this clear? Now, Down syndrome. In Down syndrome, there is a gain of extra copy of 21st chromosome. Now, this occurs due to non-disjunction of autosome. Now, non-disjunction would take place due to the failure of anaphase. Do you see that? Now, next is, so it needs to extra chromosomes. So in all, the person will have 47 chromosomes. So failure of cytokinesis after telophase can lead to the increase in the whole set of chromosomes known as polyploidy. Is that clear? Is that clear? So see, these are the symptoms of Down syndrome. Broad flat face, big and wrinkled tongue, congenital heart disease, flat back, loops on the fingertips and palm crease. Clear to all. Then these questions are asked every year. See, it is due to the trisomy of 21st chromosome in the name of the scientist Langdon Down, Broad Palm, all these studies. And then physical, psychomotor, and mental development is retarded. This was a statement based question. This was a statement based question. Then we have Klinefelter syndrome, where we have additional copy of X chromosome. So males are supposed to be XY, but instead of XY, if they gain extra X, this is what what makes them suffering from Klinefelter syndrome. Again, the resultant of total chromosomes would be 47. Overall masculine, but some feminine development takes place, like gynecomastia, which is the development of breast, and overall they are sterile. Now, they have not asked question on Turner syndrome in few years. So again, 2026 can be about Turner syndrome. Let's revise. In this, instead of female having XX, she loses that one X and becomes X0. So instead of being XX and having 46 chromosomes, she has one less X chromosome. So overall total number of chromosomes becomes 45. So such females are also sterile with the rudimentary ovaries. Root ovaries are non-functional, and there are lack of some secondary sexual characters also. So she's bit feminine, she's more masculine in appearance. So guys, with this we complete the chapter of genetics as well. Please let me know how many of you have been with me since beginning. Yes, please. How many of you have been with me since beginning? Let's take a feedback. Yes, please. How many of you have been with me since beginning? सोना नहीं है. You can't sleep like that, guys. You have to complete the syllabus. You have to complete the syllabus. How can you sleep without completing it? Yeah. So let's take second chapter of genetics. Yes, second chapter of genetics will be dealt separately because I think I forgot to download it and it's not even opening properly. Let's come on to chapter number 3. Let's come on to chapter number 3. Let's do one thing. When I take a unit wise revision of genetics, then I can revise this NCERT chapter also along with. Is that clear? Okay. All right, guys. Now coming on to the chapter number 3, that is reproductive health. See, quickly read. According to WHO, reproductive health means total well-being in all aspects of reproduction. Are these points clear? Then, what does which was another question? So another question was about amniocentesis, that it is usually done between 14 to 16 weeks of pregnancy and it is a prenatal sex determination. Now there is a statutory ban on amniocentesis in India because people were misusing it for checking the sex of the baby, and it was leading to the imminence of female feticide. That is why there was a statutory ban. Is that clear? India has its own silly problems. Then, talking about the actual medicinal or the diagnostic value of this procedure, it is actually used to test the presence of certain genetic disorders. Which one? Like Down syndrome, we have just read, hemophilia, sickle cell anemia. They can all be detected before the birth of the child. Is that clear? And then it can be decided whether we want to survive, we want to carry on with the pregnancy or not. Are these points clear? Then, Saheli is a new oral contraceptive drug which was developed by, guys, the scientists of CDRI Lucknow. Is a clear Central Drug Research Institute. Now, how does the Saheli work? It blocks the estrogen receptors in uterus and thus prevent implantation. This was 2018 question. Now, coming on to these this contraceptive. So natural methods include periodic abstinence, withdrawal method, and lactational amenorrhea. Is it clear? Now, withdrawal method is also known as coitus interruptus. In periodic abstinence, coitus has to be avoided from 10 to 17 days because high chances of ovulation are during this period, and this is a fertile period. Are this clear? In withdrawal method, what do we do? Male withdraws the penis just before the ejaculation, right, to prevent insemination. Then we have lactational amenorrhea. It is absence of menstrual cycle due to or following parturition. Why? Due to high level of prolactin. And we know when prolactin is high, FSH is reduced. High prolactin reduces the FSH. Are these points clear? Then coming on to the barrier method. Ovum and sperms are prevented from physically meeting with the help of barriers. Then one is condom. So condoms are made up of thin rubber latex sheet, right? This is for males to cover the penis. So again, to prevent insemination. Is that clear? Then the famous name is known as Nirodh. Clear? And they also prevent the person from contracting STDs and like AIDS. So there are female condoms also which are disposable just like male condoms. Both the condoms are disposable and they are self-inserted, used in complete privacy. While there are other barrier methods also, which are diaphragms, cervical caps, and warts. They are made up of rubber. They cover the cervix during the act of coitus and prevent the blocking, and they block the entry of sperms. These are all questions you can see. Is that clear? Now, instead, other than these, we can enhance the value or the effectiveness of these barrier methods by using spermicidal creams, jellies, and foams. Is that clear? They increase the contraceptive efficiency. Then there is another very important method called IUDs. Now see, they are inserted by the doctors but non-surgically. There are three different types of IUDs you must know. Non-medicated, known as Lippes loop. Copper releasing, we have different types. Multi-load is one important type of copper releasing, and then hormone releasing. The two important examples of hormone releasing IUDs are Progestasert and LNG-20. Now you must know the mode of action. See, the non-medicated ones, they increase the phagocytosis of sperms within the uterus. The copper releasing, they suppress the sperm motility and the fertilizing capacity of sperms. Is this clear? While these hormone releasing IUDs, they make the uterus unsuitable for implantation and make the cervix hostile for sperms. Are these clear? So IUDs are one of the ideal contraceptives to delay the pregnancy and one of the most widely accepted method. Other than IUDs, it is of course condoms. Now talking about pills. See, how do pills act? So pills inhibit ovulation and implantation. Also, they alter the quality of cervical mucus to retard the entry of sperms. Is this clear? So they are taken for a period of 21 days starting from first after a gap of 7 days from the menstruation, and preferably within the first 5 days of menstrual cycle we start taking it. Is this clear? Then we have these pills can be steroidal and non-steroidal. So we have this one non-steroidal pill. It is a new oral contraceptive pill. Is it clear? Non-steroidal, taken once in a week. While there are other pills which are the combination of progesterone and estrogen. Is it clear? All either we take progesterone alone or a combination with estrogen. So this is in case of pills also, as well as in case of injections, as well as in case of implants. So pills can be non-steroidal or pills can contain only progesterone or a combination of estrogen and progesterone. Now the combination of pill can come in the form of injection which is taken once or twice in a month, and similarly in the form of an implant which is placed under the skin. See, it is placed subcutaneously. So uh, the mode of action is absolutely similar, but the effective period is much longer. Is it clear? Then very important, see, if IUDs within 72 are placed within 72 hours of coitus, they can function as emergency contraceptive as well. Other than this, we have this sterilization method or surgical method. What are the two methods? The method is vasectomy, in which vas deferens is cut and tied, and this is tubectomy, in which fallopian tubes are cut and tied. Now, very important question. After vasectomy, there is no sperm present in the seminal fluid, but sperms are present in the epididymis because they are being produced by the testes, but they are just not coming out. Is that clear? In the form of this semen. Now, similarly, after tubectomy, menstrual cycle will also continue. See, surgical intervention like vasectomy and tubectomy, we have already read about this. There are questions on the same. Now, talking about the MTP, MTP is safe Okay, it is generally safe during the first trimester, that is up to 12 weeks of pregnancy. Second trimester abortions are riskier, and then there has to be permission from two medical practitioners. See, there is this act, MTP Act, amendment of 2017. And what does the amendment say? That according to this act, pregnancy can be terminated on certain grounds within first 12 weeks. Okay, on the opinion of one registered medical practitioner. Is that clear? But if it has lasted the 12 weeks, that means if you want to terminate it beyond the first trimester, so more than 12 weeks but less than 6 months, getting it? 3 months over but not 6 months completed. In that case, we need confirmation from the two registered medical practitioners. Now it says it can be terminated on certain grounds. Now, what are the grounds? Pregnancy would be a risk to the life of a pregnant woman, or physically or the mental health. And similarly, there is a risk to the child. He can be born with a physical mental abnormality. Only then we can go for MTP. Are these points clear? Then STDs or STIs. They're also known as VDs, that is a venereal disease, or RTI. What are the examples? Gonorrhea, syphilis, chlamydiasis, genital warts, and trichomoniasis. All these are completely curable. But then there are three diseases which are not curable, and we remember it as 3H. What is that? Hepatitis, herpes, and HIV infection. Is that clear? Then, so are these points clear to all? Coming on to infertility, there are techniques which will help the infertile couple to have a baby. So all the techniques which are involved in ART, that is assisted reproductive technologies, are given here. IVF, GIFT, IUI, AI, which further includes also there is ZIFT and IUT. So ZIFT and IUT are the techniques to transfer the embryo back into the female body. Is that clear? Now, there are different techniques here, so you must know which techniques come under in vitro fertilization. See, in vitro fertilization, we have a test tube baby program. So, in vitro is always followed by embryo transfer. So, in vitro occurs by test tube baby program, and second we have ICS. Sorry, not ICSI. Yes, ICSI. So, you should be very clear. In vitro includes two things. One is test tube and second is ICSI. Is that clear? While in case of wait, while there are three different methods. Yes, okay, already written here. So it was a question in 2023. The in vitro includes two methods, ICSI and test tube baby program. While in vivo includes artificial insemination, intrauterine insemination, and then there is gift. There is gift. Is this point clear? Now see what happens during zygote intrafallopian transfer, that is ZIFT. Embryos more than 8 blastomere is placed in the uterus. This is IUT, sorry. And in ZIFT, the zygote and the early embryo up to 8 blastomere stage is transferred into the fallopian tube. Is that clear? So when zygote or early embryo up to 8 is placed in the fallopian tube, it is known as ZIFT. But if it is more than 8 cell stage, it is placed in the uterus and it is known as IUT. Is that clear? And both these are the techniques of embryo transfer. So whether you do test tube baby or you do ICSI, it has to be followed by ZIFT or IUT. Is that clear? Then coming on to gift, gamete intrafallopian transfer. What is ICSI? Again, ICSI is in vitro, where a sperm is directly injected into the cytoplasm of the ova. Is that clear? Again, followed by ZIFT or IUT. So then there are cases of, when there is low sperm count, it can be treated with AI or IUI. AI or IUI. Is this clear? Alright guys. So with this, we complete this chapter also. And now moving on to the chapter of human reproduction. Please let me know that all these points are clear to you. Please let me know. How many of you still have the energy to complete these two chapters with me? Yes, please. How many of you are still energetic, we can still study and complete class 12 biology. Yes, please. Give me a thumbs up. Quick, quick, quick. Can we still complete? I know it's time, but see, I started almost at 8, so 4 hours, and we are going to complete our class 12 biology. You can, right? Perfect, perfect, perfect. This is what I wanted to hear. All right, guys. Coming on to male reproductive system. Coming here. Okay, testes. These are placed in a special pouch called scrotum for maintaining the low temperature. How low? 2.2 to 2.5 degrees Celsius lower body temperature. Why? Because this low temperature is required for spermatogenesis. Is this clear? Now, each testes is covered by a dense covering, and each testes has 250 compartments known as testicular lobules. Are these clear? Now, each lobule has 1 to 3 seminiferous tubules. Is it clear? And seminiferous tubules are the unit of testes. Now, every seminiferous tubule has two types of cells: germ cells, male germ cells, and Sertoli. Sertoli cells. Are these points clear to everyone? Now, male germ cells undergo meiotic divisions leading to the sperm formation, while Sertoli cells provide nutrition. These are the two main functions. Is that clear? Then, outer space is known as interstitial space. Interstitial space has interstitial cells. They are also known as Leydig cells, and they are known to produce androgens. Are these points clear to all? Other than Leydig cells, the other cells are immunologically competent cells present in the interstitial space. Clear? Then the male accessory duct. See, you have to remember the pathway. The pathway has been asked multiple times: 24, 19, 2023. So seminiferous tubule in the testes, then leading to leading to rete testes, then rasa differentia. See, I have labeled 1, 2, 3 for you. Then epididymis, then vas deferens, then ejaculatory duct, and finally urethra. Now, it is saying that vas deferens receives a duct from the seminal vesicle and opens into urethra as a ejaculatory duct. So basically, ejaculatory duct is formed by vas deferens plus duct of seminal vesicle. Are these points clear to all? See here. Now, the external genitalia of males is penis. This is external genitalia for erection to facilitate. It has a special tissue for erection to facilitate insemination. So, this is the function of penis, right? Covered by skin known as foreskin. The enlarged part of penis is known as the glans penis, and this is covered by foreskin. Are these clear to all? Now, there is a confusion which might occur in this paragraph that I'm going to talk about. So, male accessory glands. See, male accessory glands are paired seminal vesicle, prostate, and bulbourethral glands. Clear? Now, the secretion of all the three glands is known as seminal plasma. Now, the main constituents of seminal plasma are one, fructose, then second, calcium, and third, enzymes. Now, particularly bulbourethral secretes a lubricant for the lubrication of penis. But the question is that seminal plasma is secreted by which gland? So, all the three glands will produce semen or seminal plasma. Are these points clear? All right. Moving on to the female reproductive system. So we understand there's a pair of testes, then these are the ducts, and then we have the external genitalia, which is vulva. Is that clear? All present in the pelvic region. Now, unlike male reproductive system, which is partially extra-abdominal, female reproductive system is entirely intra-abdominal. We don't have any extra compartment for any sake. Is that clear? Now, coming on to wait. Okay, coming on to next page here. See the fallopian tube is 10 to 12 centimeter long and it has three parts: funnel shaped infundibulum, which is near to the part closer to the ovary. Is this clear? So it is closer to the ovary. Second, and infundibulum has finger-like projections called fimbriae, and which function of fimbriae is to collect the ovum from after ovulation. Is this clear? Is this clear? So the three parts of fallopian tubes, as you see here, infundibulum, then ampulla, and then isthmus. This was asked in 2024. Then the uterus, the dome-shaped part of the uterus is known as uterine fundus. Clear? Done. Now uterus single, also known as womb, then supported by ligaments to the attached to the pelvic wall, opens into the vagina through the narrow cervix. The canal of the cervix is called cervical canal. Vaginal canal and cervical canal will together form the birth canal. Are these points clear? Together form the birth canal. Now the walls, three layers of the tissues. Outer one is perimetrium, middle one is myometrium, and the inner one is endometrium. Are these points clear? Are these points clear to all? Endometrium shows cyclic changes. Myometrium is responsible for strong contractions known as labor. Now see, this paragraph is so important. Entire paragraph was in the form of match the following. So what is mons pubis? It is a fatty cushion or fatty tissue beneath skin and covered with pubic hair. Labia majora has a fleshy folds, is that clear, which surround the vaginal opening. Minora, it has again paired folds under the labia majora. Now the opening of vagina is covered by a membrane which partially covers the vagina is known as hymen. Clitoris is a tiny finger-like structure present at the junction of upper two labia minora. Is this clear? And then hymen is broken during the first, often broken during the, it is not always, it is often during the first coitus. Can be broken due to first jolt or sudden fall or jolt, and that is, or these exercises, that is why it is not a reliable indicator of virginity. Are these points clear to everyone? Now, very important, you must know the passage, the correct pathway of milk, components of mammary gland. See, so first of all, in the breast tissue, we have 15 to 20 mammary lobes. Lobes consist of many alveoli. So this is where milk is secreted. So after getting secreted, an alveoli, it goes to the mammary tubules, then into the mammary ducts, then gets stored in. This is where it is stored in the mammary ampulla, and finally it comes out in the lactiferous, through the lactiferous ducts into the nipple. Please let me know that these points are clear. This is a very important question. Very important question. So see here. Important lines. Spermatids are transformed into sperms, and the process is known as spermiogenesis. Sperm heads are embedded in Sertoli cells, and finally release of sperms from seminiferous tubules is known as spermiation. This term spermiation is very important. Now see the sequence of events. First, that is hormonal regulation. So GnRH released by hypothalamic hormone. This stimulates the anterior pituitary to release two gonadotropins, LH and FSH. LH acts on the Leydig glands and stimulates the production of androgen, while the FSH acts on the Sertoli cells and stimulates the secretion of factors which help in spermiogenesis. Are these points clear? Then talking about the structure of sperm. So what does head contain? Head contains the haploid nucleus, and it also contains a cap-like structure called acrosome. Acrosome contains these enzymes which help in fertilization of the ovum, particularly penetration. Middle piece has lot of mitochondria which produces energy, and then the tail which facilitates the sperm motility. Are these points clear? Now overall there are around 200 to 300 million sperms produced during a coitus. Okay, now these many sperms are needed for normal fertility, out of which 60% should have normal shape, and see 60 SS, remember 60 should have normal shape and 40% should have normal motility. Are these points clear to all? Please let me know. Please let me know. So seminal plasma along with the sperms is known as semen. Clear to all? Now see, unlike spermatogenesis which starts at puberty, the process of female gamete formation, which is known as oogenesis, it starts during the embryonic stage. Very important, very important. Isn't that so? And now the precursor cells or the ovum mother cells are known as oogonia, and each fetal ovary is filled with million oogonia. Is it clear? No more oogonia are added or formed after the birth. Now see, this paragraph is important. First division of prophase 1 that starts before birth, okay, and gets temporarily arrested at that, and that stage is called primary oocyte. Now, oocyte is surrounded by a layer of granulosa cells, and the stage is known as primary follicle. Is this clear? Now, some of them they degenerate, and by the time we reach our puberty, only 60,000 to 80,000 are left. Now, 2023 has asked the primary follicles get surrounded by many layers of granulosa, nucleus, and this is now known as secondary follicle. Now, secondary follicle will transform into tertiary follicle. It will have a fluid-filled cavity called antrum, and then it will grow in size and width and complete its first meiotic division, and there is unequal division here, and now it forms a secondary oocyte and a first polar body. Please let me know that all these points are clear to you all. Now see guys, the secondary oocyte retains the bulk of rich cytoplasm, while the first polar body doesn't have that. Any polar body will not have enough cytoplasm. And these points clear. Now the tertiary follicle changes into the mature follicle called the graphene follicle. Now the secondary oocyte forms a new membrane. So basically, the secondary oocyte is going to secrete this membrane called zona pellucida. So when graphene follicle ruptures, the secondary oocyte, the release of secondary oocyte along with the polar body, this is known as ovulation. This is known as ovulation. Now this whole schematic representation, that what are the phases, when does each process happen, whether it occurs simultaneously or discontinuously, this is all come up in the exam. So numerical based questions are also coming from this topic. So we see that one spermatogonia will give rise to one primary spermatocyte, two secondary spermatocytes, four spermatids, and four sperms. So ratio is 1 is to 2 is to 4. Is that clear? Then here one oogonia will produce one primary, one secondary, and eventually one ovum only. Is that point clear to all? Please let me know that these points are super clear. Now, coming on to the menstrual cycle, guys, the reproductive cycle in non-primate females is known as estrous cycle, while the reproductive cycle of primate females, is known as menstrual cycle. Who all are the primates? Monkeys, apes, and human beings. First menstruation is known as menarche, okay, and it starts at puberty. Overall, it is a cycle of 28 to 29 days. Is that clear? Okay. Now this graph is again important. You have to see the labeling properly and how each hormone gets fluctuated during each phase of menstruation, and what are the four different phases of menstruation. So there is menstruation, follicular phase, ovulatory phase, and then the luteal days. You have to see the duration of the days. So 1 to 5, right? Then this is 6 to 13th day. Then we have ovulation between 14th to 16th day. This is the shortest phase. And then 17th to 30th, we have the luteal phase. So you must know the labeling of this graph. Then lack of menstruation may be indicated of pregnancy. It may be. The answer is maybe. Always pregnancy? No. Why? Because it may also be caused due to some diseases, like diseases also, stress, poor health, and even diseases. Are these points clear? So whether absence of menstruation is always indicated of pregnancy? No, it is not. Now, similarly, we have hormonal regulation in females, same GnRH. GnRH from hypothalamus will stimulate anterior pituitary to secrete LH and FSH, which increase during the follicular stage and stimulates the follicular development. Is that clear? Now, FSH will eventually result in the higher production of estrogen because of growing follicles also. Now, both LH and FSH will attain a peak level about 14th day. This is known as LH surge, and LH surge is responsible for ovulation. This is responsible for ovulation. Are these points clear? Are you able to see how many times questions have come up from this topic? Are you able to see that? Now see, endometrium is necessary for implantation of the fertilized ovum. So it is important that this endometrium is well developed. So who's going to produce develop or generate endometrium? It is estrogen. But corpus luteum, which produces a lot of progesterone, it maintains it. See, endometrium is formed by estrogen, but it is maintained by progesterone. Are these points clear? So that is why, see, during pregnancy, all events of menstrual cycle stop and there is no menstruation. In absence of fertilization, corpus luteum degenerates, degeneration of endometrium takes place. So cyclic menstruation is an indicator of normal reproductive phase and extends between menarche and menopause. Are these points clear? So the motile sperms swim rapidly, pass through the cervix, enter into the uterus, reach the ampullary region. So this is where this is the ampullary region. This is the site of fertilization. Site of fertilization. Are these points clear? Now, fertilization will only occur, guys. It will only occur if both sperms, this is a PYQ already written here, say. See, so it can only occur if both ovum and sperms are transported simultaneously to the ampullary region of fallopian tube. Process of fusion of sperm and ovum is fertilization. And now during fertilization, sperm first comes in contact with the zona pellucida. So this is where the receptors for the sperm binding in mammals is present. Is this clear? Then, uh, so after the sperm entry, see, sperm entry induces. This means sperm entry. Sperm entry induces the completion of meiotic division, meiotic tube division, and form and meiotic division of secondary oocyte and form ovum. Again, this secondary meiosis is also unequal and it comes along with the formation of secondary polar body. Are these points clear to everyone? So see, next important topic is placenta, which acts as an endocrine tissue, produces many hormones like HCG, HPL, estrogen, progesterone. So, and also relaxin. See, relaxin also comes from corpus luteum of ovary. Is that clear? Now, see, these lines are very important. So during hormones which are produced only during pregnancy. So HCG, HPL, relaxin produced only during pregnancy. Is that clear? While there are other hormones like estrogen, progesterone, cortisol, prolactin, thyroxine. They are increased during the during the pregnancy. Is that clear? All right. So as with this, we move on to the last very important part. So fate. What are the major features of embryonic development? So you must know after 1 month, heart is formed. This was asked last year. And the sign is by hearing the heart sound through stethoscope. Second month shows formation of limbs and digits. This was asked in 2024. Then third month or 12 weeks or first trimester, major organ systems are formed, limbs and external genitalia formed. And then fifth month shows the first fetal movements and appearance of hair. Now this whole paragraph was in 2024 and 2025. Now parturition finally, that is the childbirth, occurs by fetal ejection reflex. Is it clear? So all the signals of parturition they originate from the fully developed fetus and placenta. So what are the two signals, what are the two tissues from where signals appear? One is the placenta and second is of course a fully developed fetus. These signals are collectively known as fetal ejection reflex. Now basically parturition is going to take place because of the neuroendocrine mechanism. So basically the signals arrive, right, and the posterior pituitary, it releases oxytocin. See, maternal pituitary, posterior pituitary releases oxytocin, which acts on the uterine glands, strong uterine contractions, which further increases oxytocin. So they show positive feedback mechanism. Is it clear? And then stronger contractions will lead to expulsion of baby through the birth canal. Are these points clear? So in case the contractions do not occur on their own, so what injection is given by the doctors? They inject oxytocin. Is that clear? Then coming on to lactation, that is production of milk after the pregnancy. At the end of the pregnancy or after the birth of the baby, this is lactation. So the first milk is obviously colostrum, which contains several antibodies and develops resistance of the newborn against diseases. Breastfeeding is important and during the initial period, which is highly recommended for bringing up a healthy baby. So guys, quickly, last chapter, and we're going to complete class 12. Quickly, give me a thumbs up to all of us. Last chapter of class 12, last chapter. Quickly, give me a thumbs up. Thank you, Joel, sorry. Thank you, Joel. So we are discussing important terms here, guys. So first of the important term that has appeared in the last 10-year uh, NEET exams. So see, the proximal end of the filament is attached to the thalamus. This was in 2016. This was in 2016. Now a typical embryo, see, a typical anther is bilobed, two theca, that is dithecous, right? It has four microsporangia, so tetrasporangiate. Is this clear? And it has four corners, so it is tetragonal also. Now, microsporangia will later become the pollen sacs, and they are, yes, and they will form pollen grains. Are these points clear? So, a typical microsporangium or a pollen sac is surrounded by four wall layers: epidermis, endothelium, middle layers, and tapetum. So, outer three walls, they help in dehiscence of the anther and provide protection. These are the two functions of outer three layers. While tapetum, the function of tapetum is nourishment, and for the nourishment, it has dense cytoplasm and more than one nucleus. More than one nucleus. So multinucleated condition. Is that clear? So binucleated or polynucleated condition increases the nourishing efficiency. Are these points clear to all? Anyone with any doubt in here? Now see, basically pollen sac or microsporangium is filled with sporogenous tissue, which later on gives rise to pollen mother cells. And these pollen mother cells, through meiosis, will form four microspores. Cluster of four cells. This is known as microspore. Tetrad process is called microsporogenesis. Now, because it is meiosis taking place, this is 2n, and the resultant microspores are haploid in nature. Are these points clear to all? Are these points clear? So each microsporangium has thousands of microspores. Clear to all? Now, pollen grains represent the male gametophyte. Very important. They represent the male gametophyte. Now, a typical pollen grain has two layers. One is exine, which has sporopollenin, which is the most resistant organic compound. No enzyme and no high temperature or acid or base can destroy it, right? And this leads to fossilization. This helps in the fossilization also. So preservation of pollen grains as fossils is because of sporopollenin. Are these points clear? Now, see, okay, so pollen grain are mostly shed at two cell stage. In over 60% angiosperms, it is shed at two cell stage, but in rest of them, they are shed at three cell stage. So the three celled pollen grain, it is representing two generations, one within the other. This is an important line of NEET. Is this clear? Then coming on to the pollen viability. See, in wheat and rice, the viability lasts for only 30 minutes. But for these three families, that is Sorrel is Solanaceae, Leguminosae, and Rosaceae, it is going to be for few months. The question has already been asked. So these pollen grains can be stored in liquid nitrogen at minus 96 degrees Celsius. So this method is going to increase the pollen viability. This is going to increase the pollen viability. So this is done in pollen banks or seed banks, and this is an important step of crop breeding program. Are these points clear? Are these points clear? Then the ovule is known as megasporangium. This was a question of 2016. Now the body of the ovule with the funicle is attached at hilum, and hilum represents the junction between ovule and funicle. Now, the whole ovule is filled with the cells called nucellus, which have abundant food reserve material. Is it clear? Now, so one of the nucellus is going to become an embryo sac. So basically, embryo sac or female gametocyte will be placed within the nucellus. Is this point clear to everyone? Now, these steps are important. Similar to pollen mother cell, here we'll have megaspore mother cell undergoing one meiosis and three mitosis to form mature egg embryo sac. Is that clear? Now, the embryo sac. So, how many meiosis are going to take place during microsporogenesis or in the formation of male gametes? And similarly, in the formation of female gametes. All this has to be remembered carefully. Are you able to understand? Are you able to understand, guys? So coming on to the formation of embryo sac. So see, embryo sac. So there is one chalazal end and then there is this micropylar end. Remember, micropylar end is always known as the upper end and chalazal end is always the lower end. Is it clear? So antipodals towards the chalazal end. How many antipodals? 3. And what is the ploidy? It is N. This was asked in 2023. Then polar nuclei in the center. Two polar nuclei are in the center forming the central egg, and again each with N haploid ploidy, right? Then towards the micropylar end we have these two synergids and one egg cell, and together this one egg cell and two synergids form the egg apparatus, each haploid in nature. Now synergids are characterized by the presence of filiform apparatus. This filiform apparatus is meant to guide the pollen tube. See, this is what I have already discussed. This is already discussed. Is this clear? Now, very important. The cleistogamous flowers are always autogamous. There is no chance of cross pollination. While geitonogamy, the definition was in 2023, from pollen from anther of one flower to the stigma of another flower, but the plant remains the same. Another flower, but same plant. Is this clear? While in xenogamy, there are two different plants involved and it brings genetically different types of pollen grains. Is this clear? Now, in both wind and water pollination, there are uncertainties, so we need enormous amount of pollen grains to ensure the pollination. So these lines are very important. See, pollination by wind is more common among the abiotic pollinations. So abiotic pollinations occur because of wind and water. So out of wind and water, wind pollination is more than water pollination. Is that clear? Then wind pollination is seen in wind pollination often have single ovule. It is seen in corn cobs. Is that clear? So corn cobs, the ears used here, which wave in the wind to trap the pollen grains. Then this line, pollination by wind is quite rare in flowering plants and limited to only 30 genera, mostly monocotyledons. Also, it is going to occur. It is seen in lower plants like algae, bryophytes, and pteridophytes. That is why they were called, bryophytes were called amphibians. So water was important for their pollination. Then water pollinated plants are Vallisneria, Hydrilla, and Zostera. So because they are water pollinated, they don't need to be colorful or they don't need any nectar. Now, all aquatic plants will not be water pollinated. For example, water hyacinth and water lily, they are pollinated by insects and wind. They are insects by winds. See, pollen grains in many species are long and ribbon-like. They are carried passively inside the water. They are carried passively inside the water. And see, in most of the water pollinated species, pollen grains are protected by wetting by a mucilaginous covering. Now, both wind and water pollinated flowers are not very colorful and do not produce nectar. Do not produce nectar. Are these points clear to all? Are these points clear to all? Another 5 minutes, guys. Another 5 minutes, and we are going to complete this chapter. I just hope it gets loaded. Okay. So this is done. Quick, quick, quick. Alright, let's see these pages first, which have been already loaded. So, guys, these. So let's see. Okay. I hope. No. Let's come on to this last page first. So, apomixes. So, it is a process of producing seeds without fertilization. See, production of, yeah. So, when we can produce seeds without fertilization, this is known as apomixes. And apomixes is seen to occur in Asteraceae and in grasses. Is that clear? While in case of citrus and some mango varieties, there is this another process called polyembryony taking place. Are these points clear? Okay. Now, if the fruit can be produced without fertilization, that fruit is known as parthenocarpic fruit. In if yeah, that is known as parthenocarpic fruit, and the example is banana. It can also be induced by certain hormones. Is this clear? Now, talking about this line, this residual and persistent nucellus. So, if the nucellus is persistent, it is known as perisperm. It is known as perisperm. Are these points clear to all? Then, the coconut water from the tender coconut, it represents the free nuclear endosperm. It represents the free nuclear endosperm. Are these points clear? While the white part, so the white part represents the cellular endosperm. White part, the cellular endosperm. Is this point clear to everyone? All right, guys. So here we have, so what happens after post? So what are the post-fertilization changes? Synergids will be degenerated, and then, so female eggs or gamete cell will become a zygote, and the central cell will become a primary endosperm cell. So polar nuclei will become the primary endosperm cell, and central cell will become a primary endosperm. Sorry, I told you the opposite. Wait. Yes. So basically, polar nuclei, the two polar nuclei will become primary endosperm nucleus, and the central cell will become the primary endosperm cell. Is this clear to all? Is this clear to all? Then zygote, then this pro-embryo, then globular embryo, then heart-shaped embryo, and then the mature embryo. So this stage of embryonic development should be clear to you all. Is that clear? Now, double fertilization is a characteristic of angiosperms where there are two male gametes. Two male gametes, one is going to fuse with the female gamete egg cell and form zygote, and the other one will fuse with the central cell and form what? PEC, primary endosperm nucleus or the cell. Is this point clear? Is this point clear to all? So then the technique of artificial hybridization, what are the steps? We have, first of all, we are going to emasculation, followed by bagging of the stigma to prevent undesired pollination. Then yes, then we collect the pollen grains and we debag the stigma, and then debag. After debagging, we are going to brush the stigma with the desired pollen grains, and finally we are going to rebag the whole thing. Is this point clear? These are the steps. So emasculated flowers have to be covered with a bag of suitable size. Why? To prevent the contamination with unwanted flowers. This was in 2022. So guys, with this we complete our chapter. There are 2-3 pages which are not getting loaded. Alright, so thank you so much, guys. With this, we have completed our class 12 in NCERT today. We have completed all the chapters of class 12. Now, from tomorrow on, we are going to continue with our unit wise revision, and make sure those of you who have still not started with the mock test, all of you must enroll in this series. The link is given in the chat box also. So click on the link, and every day 2 to 5 p.m. this time should be reserved for mock test. No matter which series or which session is going on, you can always watch it in the recorded form. But mock test series is essential before your preparation. Is that clear? Thank you so much, guys. Have a very good night. I am sure we are all tired, of course, है ना? But we will have a very sound and peaceful sleep, thinking that we have complete class 11. We have revised complete class 12 NCERT book. And now see, we have completed it within, see, how much time? We started almost 8, 9, 10, 11, 12, 4.5 hours. Now, if you want to revise it again, you can still do that by just increasing the speed to 1.5 or 2, and you will be able to complete it within 2 and 2.5 hours. Just 2.5 hours to complete the entire class 12 NCERT once again. So we still have time. We can still do that again. So before doing that, make sure that you sit for the mock test so that you realize what are the mistakes that you are making. So thank you so much, guys. My throat is hurting, but I'm feeling very happy that we have done this. Thank you so much, and see you tomorrow at 5 p.m. Bye bye.